हो गया एक ही एक्सरसाइज कर लेंगे ना आगे जाएगा हां मुझे उसके बाद और वाला नहीं आया मुझे आ गया ये 1/2 ऐसे ये 7 वाला हां बट कहां ये 100 वाला है ना ये आउटपुट आपको सब जिसमें ये वाला नहीं आया वो वाला जीरो आया मैं पीछे आंसर देखो तो जीरो वाला मैंने कहा क्या पता है वो वाले का मैंने उल्टा आपका इक्वेशन से निकाला था x = 0 और दूसरा नंबर से हमने निकाला 2x - 2 पीछे को गया ऐसे ही रहेगी वो 20 का है वो नहीं करेगी अगर वाला नहीं किया मैंने बहुत सारे करते हैं तो एक बार वो 20 वाला भी था लास्ट वीक वाला याद है क्या नहीं यार ये मैंने लास्ट मोमेंट पे ओके कहां से मैंने आंसर देखा और उससे उल्टा इक्वेशन निकाला ये सॉल्व कर है जैसे विदाउट सॉल्विंग ठीक है ना थोड़ा तो प्रूव करना पड़ेगा ना एक क्या देखेगा वो इतना होमवर्क कर रहा है उसमें से एसक्यू को नोटिस करेगा अच्छा हफ्ते में कभी एक दो बार कभी ना
शादी जैसे मेरा की डॉक्टर का मजा आता है बस के में छोड़ दिया वो देख के बोलता है करेक्ट सुबह सुबह अरे वो तो मेरा आदमी छोड़ दिया था
चाय की नशा की है उसने
मैं वही से
कौन सा प्रोजेक्ट आज का क्या करना क्या
ये रिसर्च पे करना है।
अरे भाई सब लोग बोल रहे हैं एक टाइप का करना है
रिसर्च पे करना है।
सुधार में तो आपने सुधार में तो स्कूल से हमारा
बता प्रोजेक्ट में
I did not write behind.
मेरे आंसर में बहुत सही और नंबर दिया था आपको आंसर थोड़ी गई
कितना पड़ेगा नहीं पर फिर क्वेश्चन से सारा किया रिवर टू और फाइनल
होटल घर पे पड़ी वीडियो में मुझे याद नहीं किया ऑब्वियसली और मैंने आज आर्गुमेंट नहीं किया
बता रही अरे वो बड़ा
इधर बैठ जाते हैं
इधर बैठ अरे इधर बैठ मैंने क्या किया
हाँ क्या आयुष पता है मैं साइड में था वो मेरा दोस्त है
इसको पीट दिया
गाड़ी वाले देख रहे यू डोंट इवन नो मी कुछ क्या है और खाने पूरी बात क्या आता है सी फॉर ड्रामा शट
उसके मुंह से तो जैसे बुला भाई इसको बोलना जैसे बुला मारती हो
दिखाना हां मैंने इससे भी माइनस
मैंने कुछ काम नहीं किया
मैं बोल रही हूं मैं वीडियो और ऐसे देख रहा हूं मुझे मार्क्स कट करेगा वो
फिजिक्स और मैथ्स कर दिया हां ना सिमिलरिटीज और वर्क अरे भाई तो तेरा नाम करा समझो नहीं करा
भाई मैंने
Yes, what was the homework for last class? उसको सब नंबर याद होते हैं कभी होमवर्क नहीं करते नहीं उसका नंबर कंज्यूमर प्रोटेक्शन है इसके बाद वो भी है और उतना ही एडवर्टाइज हम लोग को 4 बस रूम लिख दो सर अबे मैम ने तो हमें एक प्रोजेक्ट के लिए बोला देखो करके तो वो छापा फिर मैंने क्या किया एसएनसीजी सेंट्रल का बस जीएमपी क्या होता है जीएसपी क्या वैसा आधा तो नॉलेज हम लोग को इतना डीप में नहीं मिलेगी ना 8 9 11 अभी तो 8 9 11 तेरी ना 2 लाख को दिखा रहा
Okay, your doubt, please. Question 1-7.
So 4 6
One by one, we go in sequence. Don't go. Lecture me, of course. In the first question, question number 1, which are your doubts?
So 7.
4 6
4.
So 7.
7 बाद में, ठीक है? 4 6 also. Okay.
तो बोर्ड पे लिख लो
हाँ. और कोई सेवा नाम
सर देखते हैं सर 1 4 नहीं
मैं क्वेश्चन नंबर 1 का पूछ रहा हूँ, तुझे भाषा नहीं समझती है, बता. हमें तो संस्कृत आती नहीं, ऊपर तो लोग तो संस्कृत को. फर्स्ट क्वेश्चन में से कौन सा? 7. upon x 100 upon x plus 5 is equal to 1. We will take LCM. This gets multiplied and this gets multiplied. Okay. 100x plus 500 minus 100x. Stand up there. Stand up. Yeah, stand up. खड़ा है अभी, चुप करके, ठीक है? X squared plus 5x is equal to 500. X squared plus 5x minus 500 is equal to 0. We want two factors of minus 500 so that the difference is 5. 25 and 20. This is positive, so plus 25 minus 20. Next out of the same question. proper question. Proper question, I've seen many of you making a mistake in this sum. Form the quadratic equation whose roots are 0 and minus 8 by 9. So x is equal to 0 or x is equal to minus 8 by 9. Therefore, 9x is equal to minus 8. Therefore, 9x plus 8 is equal to 0. Therefore, x into 9x plus 8 is equal to 0. 9x squared plus 8x is equal to 0. Many of you have left it here only. This is the quadratic equation. Question number 4. Without solving the quadratic equation, 6x squared minus x minus 2 is equal to 0. Find whether x is equal to 2 by 3 is a solution of the equation. X is equal to 2 by 3 will be a solution if on substituting this value of x on the left hand side, we should get LHS is equal to RHS. So x is 2 by 3. Eight by 3 minus 2 by 3 minus 2. 8 minus 2, 6. 6 by 3, 2. 2 minus 2 is equal to 0. So that is equal to RHS. Since LHS is equal to RHS, therefore x is equal to 2 by 3 is a solution of the given equation. That's it. Next doubt. Question number 6 you all said, right? X is equal to minus 3. And x is equal to 2 by 3. These are the solutions of the equation mx squared plus 7x plus n is equal to 0. Find the values of m and n. Something similar to what we did here. Here we have to verify whether x is equal to 2 by 3 is a solution or not. Here we have been told that these are the solutions. So we write substituting x is equal to minus 3 in mx squared plus 7x plus n is equal to 0. Therefore, m into minus 3 the whole square plus 7 into minus 3 plus n is equal to 0. 9m plus n is equal to 21. This is our equation number 1. Okay. Now, substituting x is equal to 2 by 3 in the given equation. So we get m into 2 by 3 the whole square plus 7 into 2 by 3 plus n is equal to 0. 4m by 9 plus 14 by 3 plus n is equal to 0. LCM is 9. So 4m plus 42 plus 9n is equal to 0. 4m plus 9n is equal to minus 42. This is our equation number 2. We have to find the values of m and n. We can also do it by substitution. I can write therefore n is equal to 21 minus 9m. Yes or no? Which I am going to substitute here. 4m plus 9 into 21 minus 9m is equal to minus 40. Understood this? So we get 4m plus 189 minus 81m is minus 42. That is minus 77. Is this clear? So we get n is equal to 3. Substitute m is equal to 3 here. N is equal to 21 minus 9 into 3. 21 minus 18. N is also 3. Is this clear? Completely here. Over is not. This is out. Yes, bache. 27 and इधर अच्छा 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 ओ सॉरी मेरा टेबल्स बहुत हो रहे हैं ठीक थैंक यू हाँ हाँ शॉर्ट में किया क्या शॉर्ट में Question number 7. Given that 2 is the root of this equation, like what we did here, substituting x is equal to 2 and 3x squared minus p into x plus 1 is equal to 0, so that we can get the value of p. Therefore, 3 into 2 squared minus p into 2 plus 1 is equal to 0. This is 12 is equal to p into 3. Therefore, p is equal to 4. We have got the value of p. Where else can we see p? We can see p here. Substituting p is equal to 4 in px squared minus qx plus 9 is equal to 0. 4x squared minus qx plus 9 is equal to 0. Now we have to find the value of q. And we have been told that this equation, that is this equation, where I have substituted p as 4 as equal roots. Equal roots means b squared minus 4ac is equal to 0. So I must first compare with ax squared plus bx plus c is equal to 0. Therefore, a is 4, b is minus q, and c is 9. Then I'll write, since roots are equal, therefore b squared minus 4ac is equal to 0. Therefore, minus q squared minus 4 into 9 is equal to 0. Q squared is equal to 16 into 9. Therefore, q is plus or minus 4 into 3. So q is plus or minus 12. Did you understand? Yeah. Yes. So we've got the values of p and q. In the next, suppose 8 sum, right?
Yes.
We'll first solve this bracket. The LCM of this bracket is x. So this becomes 1200 plus 2x upon 2 upon x, sorry, into x minus 10 minus 1200 minus 60 is equal to 0. Now we'll take LCM. LCM again here is x. And we'll also open this bracket. 1200x minus 12,000 plus 2x squared minus 20x minus 1200x minus 60x is equal to 0. Have you understood? The LCM is x. Any doubts? This 1200x minus 1200x gets cancelled. 2x squared minus 80x minus 12,000 is equal to 0. Divide by 2. X squared minus 40x minus 6,000 is equal to 0. I want product as minus 6,000, 40, 100 and 60. Minus 100 plus 60 gives you minus 40. minus 100 into 60 gives you minus 6000. X squared minus 100x plus 60x. X into x minus 100 plus 60 into x minus 100. These are the four answers. This is clear. Time पूछेगी तो
नहीं आगे
वो तो इससे पूछेगी तो नहीं कोई पूछेगा क्या बोलता है नहीं I don't think we have done this. But I have not done this.
उधर एक लास्ट सम बाकी 3 बाकी सब हो गया.
I will do many more. मैं तो ऐसे ही पूछ लेता हूँ. आप लोगों को मैसेज दिया गया था टेक्स्ट बुक लाने को. लाए हो ना?
यस सर. तो कितने मैसेज आए?
हाँ. दूरदर्शन है ना, दूरदर्शन केंद्र वहाँ पे आया था. आपको नहीं पता? आया था कैसे? आया था. बेटा वो जो है ना तो आपको फेसबुक से डाउनलोड कर दो.
तो कुछ बच्चे जल्दी करे तो उसको यहाँ पर पता नहीं चला.
तो इसको किसने मिला? कौन बच्चे जल्दी करते हैं? क्यों गए इतने जल्दी?
और इधर से हम लोग पहले भी चले
ज्योग्राफी का लेके आते तो हम लोग मैथ रीडिंग करते उसमें. कोई एक्स्ट्रा टेक्स्ट बुक है किसके पास? गरीब को दे दो. ये तो मांगता है, मांगता कौन? Now why I wanted you to get your textbook? इसको नहीं मिला. उत्तर जी को don't worry. हमसे नहीं करेंगे. तो टेक्स्ट बुक है किसके पास? तो दोनों शेयर करें. ज़्यादा नहीं. हाँ?
कौन सा टेबल है?
वो देखते हैं. Exercise 5.3 we have not done question number 2. So we will do See, the question says solve each of the following equations for x, giving your answer correct to 3 decimal places. Whenever you get this type of a question, correct to 1 decimal place, correct to 2 decimal places, or correct to 3 decimal places, you will always use the method of the quadratic formula. Okay, the quadratic formula. So we have 3x squared minus 12x minus 1 is equal to 0. In any case, this question cannot be solved by splitting the middle term. So I have no other choice but to use a quadratic formula. So my first step in the quadratic formula is to see that this equation is in the standard form. Yes, this equation is in the standard form. I don't have to do any changes. So I will start by comparing with ax squared plus bx plus c is equal to 0. So we get a is 3, b is minus 12, c is minus 1. Okay. Our next step, using the quadratic formula. So I write down the formula. X is equal to minus b plus or minus root of b squared minus 4ac the whole upon 2a. And I shall now substitute minus of minus 12 plus or minus minus 12 the whole square minus 4 into 3 into minus 1 the whole upon 2 into 3. Any doubts? This is 12 plus or minus root of 144 minus into minus plus 12 upon 6. Okay. This is equal to 12 plus or minus root of 156 upon 6. Now 150, 2, 7, 8, 2, 39, 3, 13, 13, 1. Is this clear? That is 12 plus or minus 4 into 3 into 13 upon 6. 4 is a perfect square. So this will come out as 12 plus or minus 2 root 39 by 6. Okay till here, everybody? Any doubts? Therefore, x is equal to, taking 2 commons, we'll get 6 plus or minus root 39 by 6. Doubt. बेटा ये दोनों 2 टर्म है आपके पास, इसमें से आपने 2 कॉमन निकाला, 12 or 2. If I had given you 12 plus 2x and I told you to factorize, wouldn't you have done like this? That's what I've done. Understood? Because I cannot cancel this and this. Now I can cancel this. Is this clear? That is x is equal to 6 plus or minus. Now, in your textbook, you have these square root tables. You have these square root tables. Now, there are 2 types of tables that you have in your textbook. One is square root tables from 1 to 10. Okay? Square roots from 1 to 10. which you have on page number 446 and 447 of the textbook. ऊपर देखो हेडिंग है स्क्वायर रूट फ्रॉम 1 to 10. उसमें आप फर्स्ट कॉलम देखो 1.0 से लेके 9.9 तक है. आपके उसमें देखो जरा हेडिंग देखो स्क्वायर रूट फ्रॉम 1 to 10 because this has not been taken from the textbook. हाँ ठीक है देखा. हाँ उसके फर्स्ट कॉलम देखा आप सबने 1.0 से लेके 9.9 तक फर्स्ट कॉलम देखो. ठीक है और नेक्स्ट पेज पे आ जाओ 448 एंड 449 हेडिंग देखो स्क्वायर रूट्स फ्रॉम 10 to 100. उसमें भी फर्स्ट कॉलम देखो 10 से शुरू होता है लास्ट कॉलम 99 है होगा है ना. ठीक है तो मैंने आपको दोनों दिया है. फिलहाल हम लोग we have got root of 39. So I am going to see the page which has got from 10 to 99 because I want root of 39. So root of 39 में मैं जाऊंगा 39 के कॉलम में जाऊंगा और उसके बिल्कुल बाजू में कौन सा नंबर दिख रहा है मुझे 6.245. सबको दिखा उधर. यही अगर मुझे आता नंबर 61 तो I would have gone to the first column 61 and उसके बाजू में कौन सा नंबर दिख रहा है मुझे 7.810. यही अगर आता 96 तो I would have gone continued down and my answer would have been 9.798. Is it clear? But here we have got 39 and 39 is 6.245. So here we are going to write 6.245. Have you understood? Is this clear? Now, therefore x is 6 plus 6.245 by 3 or x is 6 minus 6.245 by 3. Any doubts? This is 12.245 by 3. This is minus 0.245 by 3. Okay. .245 divided by 3. 4. 2. नहीं डिवाइड होता है 0 डालेंगे 8 times 24. ठीक है 5 1 time 3 2. हम लोग को बोला है up to 3 decimal places. जब मुझे 3 decimal places बोलेंगे तो I will carry out division till 4 places. मुझे अगर बोलेंगे 2 decimal places तो I will carry out division up to 3 places. अगर मुझे बोलेंगे 1 decimal place तो I will carry out division to 2 places. अभी 0 6 बस मैंने कर दिया जो भी आता I don't care. So now I write therefore x is equal to 4.0816. But I want up to 3 decimal places. My 4th decimal place, if it is 5 or greater than 5, I will add 1 here. So my answer is x is equal to 4.082. Did you understand why I have asked you to go for 1 extra decimal place? Is this clear? Now coming to this one also. 0.245. तो ये वैसे डेसिमल है नहीं डिवाइड होता है तो 8 डालेंगे हम लोग 5 तो 1 सेम ही आ गया, ठीक है ना। तो x is equal to minus 0.0816, therefore x is equal to minus 0.082. Have you all understood this? Any doubts? Sure. So complete this sum and the remaining two sums also. Okay. You some copy here. You can 16 copy. No. No. A1 and excretory mutation. What is the coefficient of this?
Third sum का 5 पे तो है तो मैं 4 इसमें हो गया ठीक है
क्या
सर वो थोड़ा मैंने कहा 0.3915 है
हाँ
तो ये
0.392 5 or greater than 5. इसको माइनस कर दो, मुझे राइट माइनस प्लस 9.6 ये नेगेटिव है, ये पॉजिटिव है, आपको सब्ट्रैक्ट हो जाता है। Then you can have independent both are minus और both are plus. और ये minus से आपका क्यों होगा? अब ये प्लस वाला होगा।
So आपको नेगेटिव का
Okay, 8 minus 7.616 is not 1.6. 8.000 minus 7.616 करके देखो। गौरव करते क्यों? Has everybody finished?
Yep.
Now I want you to try this one. Please try this one. 3x squared plus 5x minus 9 is equal to 0. देखो ये वाला सम देखो, और ये अपने को इधर मिला 133 मिला, तो अर्पिता जी ने इसको फैक्टराइज किया और ऐसे लिखा बिल्कुल सही है। और फिर इन्होंने क्या किया, इन्होंने रूट ऑफ 19 का वैल्यू लिया टेबल में से। रूट ऑफ 19 shown as 4.359. Then she wrote root of 7 as 2.646. है ना? लाइफ कितना डिफिकल्ट हो गया, पिल्ले की तो आंखें ऐसे हो गई, देखो ऐसा कितना डिफिकल्ट हो गया, तो हम ऐसा नहीं करेंगे। Whenever you get a number, because this type of question is not given to you in the volume, but it is there in the textbook, therefore I have taken this sum. See, till now whatever numbers, the तीनों sum जो किया हमने, जो root के अंदर जो number आया था, it was less than 100. So we were using the table from 10 to 99. But now, what about the table from 1 to 10? This is where we are going to use it. So I'm going to write this as 1.33 into 100. हम लोग अगर इसको 100 मल्टीप्लाई करेंगे तो ये 133 हो जाएगा। So I have now rewritten this as 1.33 into 100. Why I have written this as 1.33 into 100? Because 100 is a perfect square. And I can write my next step as -5 plus or minus 10 into root of 1.33. Have you understood this? Now come to your table from 1 to 10. Square roots from 1 to 10. अपने पास नंबर क्या है 1.33. So I will go in my first column 1.3. हाँ, और ऊपर जो हॉरिजॉन्टल लाइन में 3 है और एक, क्योंकि 1.33 है, उसमें मैं नीचे जाऊंगा तो what is the number that I see there? 1.153. 1.153. Did you see it there? समझा क्या 1.33 का? पहले समझा तो मुझे बताओ 2.87 का क्या होगा?
So 2.87 or 1.695.
हाँ, समझ गए सब लोग? और 7.35 का क्या होगा? 2.711. अभी समझ गए ना ये? हाँ, तो हमारे पास नंबर क्या था 1.33, तो ये 1.153. Is this clear? So now this will become -5 plus or minus 11.53 because I have done the multiplication with 10. Have you understood this? Is this clear? And then the normal procedure will continue. Please complete this. Okay. This is wrong. This one is wrong. You go. भगवान अगर मिस्टेक करते हैं क्या क्या होता है? अच्छा आपने उसको 2 पे कर दिया, 2 बेसिस पे कर दिया, 3 पे करेंगे तो ऐसे आएगा। और इधर आपने रॉन्ग किया है, ये जरा चेक करो आपका माइनस साइन किधर जाएगा, ये वाला। हाँ अभी आपका रॉन्ग है, साइन किधर है, साइन चेक करो। अगर 3 में बोलूंगा तो कहाँ पे पहले वाले को रोक सकते, अगर मैं बहुत 3 डेसिमल है तो 4 डेसिमल दिखाएगा, अगर 3 करना हो तो आपका लिखोगे, अगर मैं सिर्फ 3 डेसिमल प्लेस चाहिए क्या लिखोगे? Are you sure? It's 9 होगा। ये वाला निकाल रहे हो तो सेम रखो, हम एक ऐड करेंगे और चेक करेंगे। नहीं देखो ये 5 से नहीं हो रहा डिवाइड, यू शुड हैव पुट 1.0 0 आते हैं नहीं, मैंने एक जगह पे डाल दिया है इसे कैलकुलेट कर हो गया सबका? Solve. See what we'll do in the given form, it is not a quadratic equation. So we'll write it like this. This is clear. And then we'll carry out substitution. Let x squared be equal to m. Therefore, m squared minus 2m minus 3 is equal to 0. We want two factors of minus 3. One is negative, one is positive. So that the sum is minus 2. So minus 3 and plus 1. M squared minus 3m plus 1m minus 3 is equal to 0. M minus 3 plus 1 m minus 3 is equal to 0. One over 1. Continue.
Sir, m is equal to minus 1 or m is equal to 3. तो m is equal to 3 हुआ क्योंकि x square is equal to x square is equal to 3 and m x square is equal to minus. So minus 1 का रूप में निकल सकता तो
But square of a number
is never negative.
is never negative. Therefore, discard it. Is this clear?
Yes.
Next step, bolo.
So to x square is equal to 3. So x is equal to 1.7.
Plus or minus
Yes.
root 3. Have you understood this? Clear, everybody? So this is from your homework questions. Question number 4, sub question number 1. You please also do sub question number 2 now. Okay. Total meaning. ये सेम What do you think nonsense? कितना आंसर है प्लस माइनस कौन हाँ
प्लस माइनस 3 और प्लस माइनस 1
हाँ बच्चे हाँ वो भी मतलब होता है ना Thank you very much.
That's my big job. पांचवा छोटा वाला है ये सारे मैं रात को सही कर लाइट ही ना उस वक्त याद है लाइट ही नहीं पता ना ये तो है कितना अबे घटिया किस्मत थी इन लोगों का सही था इसलिए कर रहे हो हम लोग को बढ़ना पड़ा आगे नहीं क्यों हुआ अरे हम लोग अपना नहीं काम में नहीं करते हैं मैं तो इसको लिख कर दिया
दोनों की इंग्लिश की टेस्ट रहती है नहीं बस पढ़ेंगे सुनो क्या ये बात संडे को आपकी
नहीं है
लीनियर एलिमेंट जिसको आना चाहिए सिमिलैरिटी का नहीं होना चाहिए कब नहीं हुआ लीनियर इसको अभी तक कंफ्यूजन है ना होगा अरे तो क्या फर्क पड़ता है बाकी तो समझ सकते हो उसका मतलब नहीं पागल क्या भाई दिमाग खराब होगा हिंदी टेस्ट तो किया
अरे तक
नहीं है तो सारे है ना अब मैं ये संडे हिंदी का टेस्ट सीरीज है
नहीं आता टेस्ट
क्या है मेरी बात कर इसलिए नहीं लगवा रहा अच्छा बहुत हैप्पी हो रहा है ये तुम लोग की क्लास में अपन शांति वगैरह above 60
above 60 कौन क्या है बताओ
above 60 है
above 60 कौन क्या बताओ उसे मैं काट दूंगा कोई कह रही है
मुझे 60 मिला
कितना काटते हैं लोग
अबे हम लोग मेरा
हिंदी में आया नहीं आया मुझे फर्क ही नहीं पड़ता
अरे क्या बोल रही हो बस 7 मिनट में मेरी क्लास बोल रही है
नहीं पर रिबन में ही दिया ना शायद
अरे रिबन में ज्यादा मार्क्स देना
वही बात
क्यों डिस्कस कर रहा है हिंदी वाला
मुझे 41 मिला
बचपन से जान से काट
कभी भी मैंने ऊपर ही रहेगा
आई नीड टू स्टडी कॉमर्स एंड साइंस भाई पे कॉमर्स से बच्चे
चेंज ऑनलाइन में गेम खेल रहे हैं सॉरी मैं नहीं खेल रहा नहीं भाई पागल है पता नहीं क्या बात है
क्या है बढ़िया वाला खाया मैंने कभी टेस्ट नहीं किया
नेशनल पार्क तक यार हिंदी वाले दो दिन है मन में आराम दे देते हैं अभी हिंदी का प्रोजेक्ट आराम से चेक कर लेते मेरी बहन क्या काम में आता है पार्टी ने तो बहुत कोई और नहीं, गुप्ता हो सकता है, पार गुप्ता। अबे भाई करने के लिए कोई भी कुछ कर दे, किसी को डुबाना है तो। वैसे तो मैं शहीद का फेक आईडी बना दूं। ट्राई करना। मैं लगाऊंगा। मैं तो तेरा किस जगह का आईडी बनेगा पता नहीं चलेगा। तेरा फोन नंबर, मेल आईडी, घर का नंबर सब मेरे पास है। क्या घंटा कोई निकाले? तेरे पापा का, तेरे मम्मी का, तेरा सबका फोन नंबर मेरे पास। क्या काम है इसमें? मजा आता है।
स्क्वाट कर उस पे।
अरे यार मेरा बैग दिया करता लो।
अरे मत दे दे।
पेंसिल बेटा बैग इधर आ। अरे क्या निकाल ये साले मुंह पे मत मार। अबे कितने साल का आता पारा है? अरे बस अरे निकल नहीं रहा क्या है? खत्म हो गया। ट्राई करना, तेरा पहुंचेगा भी काबू होता है।
मैं इसको ऊपर खेलने का पसंद करता
हूँ। उल्टा कर जाता है।
साले देर में चलते हैं कुत्ते।
तेरे को क्या घंटा पूछा लड़ा होगा मेरा बहुत साले हमारी मेरी मां है ना तेरी कुछ पता हुआ तेरा पता होगा भाई सोचा कि मम्मी कहा तेरा भाई मेरी मम्मी को रात को रंडे के में रोज मिलती है मेरे कपड़े रखे यार दुकान में पूरे रोज आते हैं जो गुंडा से खाना नहीं मिलता कुछ नहीं कर बैठती भाई मेरे घर में पूरा खाना रहता है तेरे घर में खाने के लिए आके तेरे घर के पैसा भी नहीं भाई बचका गाड़ी तरीका के साथ खुला के मां की जल्दी जोर से बैठे देना भाई अबे कितनी कट जाएगी या कुछ बता देगा मुझे अरे मुझे मेरा पाउच बहुत अबे कितने आगे बैठ जाओ
Exercise 5.4, we had done the first question. That was the sum which I had solved before. Question number 2, solve. Again here, I said x squared minus 5x be equal to m. Okay. So this will become m squared minus 7m plus 6 is equal to 0. So two factors are minus 6 and minus 1, correct? M squared minus 6m minus 1m plus 6 is equal to 0. M common, m minus 6. 1 common, m minus 6. Therefore, m minus 6 into m minus 1 is equal to 0. Resubstituting. M is equal to x squared minus 5x. So this becomes x squared minus 5x minus 6 into x squared minus 5x minus 1 is equal to 0. Therefore, x squared minus 5x minus 6 is equal to 0, or x squared minus 5x minus 1 is equal to 0. Now what has happened is we have got two quadratic equations. There are three possibilities. Both these quadratic equations could be factorized by splitting the middle term, or one of them could be factorized by splitting the middle term, and the other one we would have to use the quadratic formula. Or if you're really unlucky, then both you would have to use a quadratic formula. Is this clear? So it's like one question which has now become two questions. Please complete it. Also, when you use a quadratic formula sometimes, here in this case, it's not factorizable by splitting the middle term. So you're going to use a quadratic formula. When you are using a quadratic formula, it may so happen in some cases that your b squared minus 4ac may be less than 0. If your b squared minus 4ac is less than 0, means this quadratic equation has no real roots. So it will come to an end there. Is this clear? You need not solve it further. Separately करो इसके बाद में क्या जरूरत है? ऐसा कोई रूल नहीं के साथ में करना है. Make it present with this. That's it. अभी अपने को सिर्फ सॉल्व बोला है, ठीक है ना? तो हमें आंसर आया था 5 प्लस और माइनस √29/2। क्यों आगे करेंगे? बोला है कि आपको 3 डेसिमल 2 डेसिमल नहीं बोला है, छोड़ दें बिल्कुल। कोई सरप्राइज इसने बुक लिया, अपने को बोला है कि अगर सॉल्व ऑफ 3 डेसिमल प्लेसेस नहीं बोला है, जब तक वो नहीं बोलेंगे आप नहीं कर सकते। Please do sum number 3 and 6. 3 and 6. Yes. Third and sixth. Opposite. Opposite. X is minus 3 by 2. X not equal to minus 3 by 2. We can, so we will not consider this. Equal to minus X is minus. उसमें यही आंसर नहीं आ रहे नहीं आ रहे Here we will take LCM. Okay. 4 6 are 24. Oh, you will get two values of X.
Sir, so far,
Huh?
not every.
Huh?
कुछ नहीं कर.
360 कर. So next thing Roots are equal बोला ना बच्चे तो आपको कंपेयर करना है जो भी two roots are equal don't open the brackets get the value of a b and c since roots are equal therefore b squared minus 4ac is equal to 0 उससे आपको m मिलेगा फिर वो m का वैल्यू इसमें डालने का और सॉल्व करने का यूजिंग क्वाड्रेटिक फार्मूला क्यों लिखते हैं तो m का वैल्यू मालूम नहीं since roots are equal therefore b squared minus 4ac is equal to 0 तो उससे आपको m का वैल्यू मिलेगा कितना पैसा खर्चा काफी खर्चा किया कोई जगह पढ़ा भी नहीं कोई चीज में you should be thankful ऐसे कपड़े लेके आएगा तो गंदी लगेगा। Then something that can protect you. Unnecessarily tempting also. Yeah.
Word protection.
रोस्ट करने में मेरे से माइंड बॉडी मैं भी उसी स्कूल से आया हूँ। ऑस्ट्रेलिया में बुलिंग हम लोग से सीखे। Over everybody?
Yes.
Next two sums we are not going to do. Sorry, you will do the sum number 4 we are not going to do because it has never come in the exam. But you will do sum number 5 yourself now. Sum number 5. क्या कर रहे हैं? दिखा दे, दिखा दे, दिखा। काय को मॉडल करते हैं बाबा? सर m किधर गया उधर? m होना चाहिए ना? हाँ? See, there is already x upon x plus. Let x upon x plus, you can never write it as x. You can take any other letter other than x. So you take it as m or a or y, whatever you want. Just you get it wrong. देखा सकते हो? देखा ना बेटा? नहीं आया तो पूछ लो बच्चे। अब ये -7 और 12 है ना? तो तुझे 12 के फैक्टर्स चाहिए। 4 3 और 12 होते हैं? अभी सॉल्व। ओवर from 5.3, your homework is question number 2 and 3. One here, one, 2 and 3. नहीं करके आप बाहर बैठो, ठीक है? Question number 1, 2 and 3 from exercise 5.3 homework questions. From unsolved exercise 5.4, you will do question numbers 1, 2, 3, 4, 6 and 7. Please submit. किया तो यार एक बार दे दो सर हाँ क्या ही फर्क पड़ना चाहिए ये बार बैठेगा
5.3 का 1 2 3 और M7 आ रहा है
7 डालेगा इधर तो 4x squared minus 4x plus 1 is equal to 0 minus 2x minus 1 the whole square. Now we will start with solving schedule है स्कूल में चलो अच्छा एग्जाम में क्या है अभी क्वाड्रेटिक का दोनों चैप्टर है ना अच्छा तो अभी हम लोग को क्वाड्रेटिक प्रूफ किया है सभी एग्जाम। So we will continue with quadratic equations word problems because already we have got now we will start with quadratic equations word problems. In word problems, what we are going to do is you are given certain conditions. So you are going to write these statements. Number one, remember, whenever we are doing quadratic equations, whichever quantity, whichever percent, whichever number comes second in order, generally we take that as x. For example, if I say the length of the rectangle is greater than its breadth by 3 centimeters. In my statement, breadth is coming second. So I'll say let the breadth of the rectangle be x centimeters. Therefore, its length will be x plus 3 centimeters. If I say Anil is younger than Sunil by 2 years. So I take Sunil as x. So let Sunil's present age be x years. Therefore, Anil's present age will be x minus 2 years. Is this clear? Generally, not as a rule. Only thing is, if you take the assumption in such a manner, things become simple for you. Read the question, write the statement very carefully. When I say consecutive numbers means one after another. So if I take my assumption, I start with the consecutive numbers to be x. The next one will be x plus 1. The next one will be x plus 2, x plus 3, and so on. But if I say consecutive odd numbers, then you will take them as x plus 2, x plus 4, x plus 6, and so on. If I say consecutive even numbers, I will take them as x plus 2, x plus 4. तो ये सोच रही दोनों तो सेम है, बराबर कि नहीं? कि नहीं? नहीं, तो क्या लेना चाहिए मुझे?
x plus 1, x plus 3.
अच्छा, ब्यूटीफुल। देखो बेटा, अभी आपसे मैं पूछता हूँ, लेट मी सी योर नॉलेज। 3, 5, 7. 3 plus what is 5? 5 plus what is 7? 2. So difference between consecutive odd numbers is also 2. Difference between every even consecutive number is also 2. So जब भी consecutive odd or consecutive even आएगा, so we will take them as x plus 2, x plus 4, and so on. सिर्फ consecutive आएगा, so you will take them as x plus 1, x plus 2. Difference is 1. This is clear. Clear everybody? वो गलती मत करना odd even सोच के, नहीं, चाहे odd हो चाहे even हो, the difference between every consecutive odd number is also 2, and every odd number and even number both is 2. Is that clear? So the first one, the product of two consecutive integers is 56. Let us see how we present this sum in the exam. Statements, word problems. So the statement is to be written and let the two consecutive integers be x and x plus 1 respectively by the given condition x into x plus 1 is 56. This is clear. Therefore, x squared plus x minus 56 is 0. I want the product as minus 56, sum as 1. So minus 7 and plus 8
minus 7.
This is clear. Therefore, x squared plus 8x minus 7x minus 56 is 0. Therefore, x plus 8 minus 7 into x plus 8 is equal to 0. Therefore, x plus 8 into x minus 7 is equal to 0. Therefore, x plus 8 is equal to 0, or x minus 7 is equal to 0. Therefore, x is equal to minus 8, or x is equal to 7. What's your answer? Huh? 7 or
minus 8.
wrong. When x is minus 8, I have to show like this. Then x plus 1 is minus 8 plus 1, which is minus 7. When x is 7, then x plus 1 is 7 plus 1, which is 8. Therefore, the two consecutive integers,
ऐसे word में लिखना है पूरा.
हाँ, minus 8, comma, minus 7, or 7, comma 8. Did you understand this? Is this clear?
Yes.
Any doubts?
No, sir.
Question number 2. The sum of the square of two consecutive natural numbers is 41. Let the two consecutive natural numbers be x and x plus 1 respectively. इज्जत देने का उसको रिस्पेक्टिवली मतलब वन आफ्टर अनदर, इसके लिए रिस्पेक्टिवली इज्जत के लिए नहीं लिखते ये, समझा कि नहीं? बाय द गिवन कंडीशन the sum of the squares, so x squared plus x plus 1 the whole square is 41. Is this clear? Therefore, x squared plus 2x plus 1 minus 41 is 0. Therefore, 2x squared plus 2x minus 40 is equal to 0. Divide by 2. Therefore, x squared plus x minus 20 is equal to 0. 5 and minus 4 are the factors. Therefore, x squared plus 5x minus 4x minus 20 is equal to 0. Therefore, x into x plus 5 minus 4 into x plus 5 is 0. Therefore, x plus 5 into x minus 4 is 0. Therefore, x plus 5 is 0, or x minus 4 is 0. Therefore, x is minus 5 or x is 4. What's your answer?
Sir, if x is 4, then the solution set will be 4 and 5 if x is -5. The solution set will be -5 and -5.
Absolute nonsense. Because you have not read this. But
The 4 and 5.
shut up and listen. Does not belong to natural numbers. समझा? याद नहीं है, लिखने का है खाली। तो बोलते ना पोस्ट ऑफिस के बाहर बैठे रहते हो, लिखते रहो, ऐसा ही, याद नहीं देने का। Therefore, x is 4. Therefore, x plus 1 is 4 plus 1, which is 5. Answer. The two natural, the two consecutive natural numbers are 4 and 5. That is why I did both these questions. एक इंटीजर्स वाला था जिसमें नेगेटिव नंबर्स आर एक्सेप्टेबल, एक नेचुरल नंबर्स था जिसमें नेगेटिव नंबर्स आर नॉट एक्सेप्टेबल. So that is how you are going to present yourself. Is this clear?
Okay, sir.
Complete both the questions. तू और अच्छा देख तेरा भाई बोल रहा है कुछ पीछे से.
इंटरसेक्शन नहीं आने वाला
मेरे हिसाब से तो दूसरे स्कूल में बोला है नहीं
सर मैंने वो सिलेबस का चेक किया उसमें भी
नहीं है ना, because this is one child told me it's not part of us. That's why but we have done it as such. हमने किया था तो वो I have done it with all, but when I was reading it, a child told me, that's why I came up with it. And I asked you, what is your teacher saying? Now do those who finish will now do sum number 5. क्या जगह नहीं तो एक वॉल्यूम अंदर रखो। हाँ तो रख तेरा सेहत बराबर है ना।
सब नहीं है बहुत नहीं तू बढ़िया
बोलने नहीं आता
मुखर्जी Fifth. You have to do the fifth sum, not the third sum. क्वेश्चन का भाषा पढ़ना फिर लिखना आंसर। Please write one. Please, have you written the assumption statement? Have you written the assumption statement? Read it properly. Cancel this and write it again. Read the question properly.
सर बस स्टेटमेंट
लिखना बराबर से, पढ़ना उधर क्वेश्चन क्या दिया है। रॉन्ग है, वापस देख, रॉन्ग है, पर बराबर से वापस देख। समझा तू, तेरे को क्यों बोलता है, लेकिन देख ऊपर, ऊपर भी देख। You lose marks otherwise. मुखर्जी खड़ा रहे, तो खड़ा रहे ना, खड़ा बहुत नाइंसाफ है, जरा कंप्लेन कर लेना। तो पुलिस चौकी के एड्रेस को बताया था बोला, ज्यादा दूर नहीं, 3 टाके की तरफ से उधर से अच्छा वड़ा पाव भी मिलता लेके। Let me see. No, you're not writing the statements properly. Please write the assumption statement properly again and then bring it to me. Read the question. Read the question properly and write the statement properly and then show me your answer. You don't have to rediscover anything. You have to simply copy what is given in the question as your statement and as your answer statement. Question number 3. The sum of a number and its reciprocal is 4.25. So it's very simply you say, let the number be x. Therefore, its reciprocal is 1 by x. By the given condition, x plus 1 by x is 4.25. Question number 4. The square of a number added to 1/5 of it is equal to 26. Again, let the number be x. By the given condition, it says square of a number x square. ठीक है? Added to 1/5 of it, x by 5 is equal to 26. Complete this, please. Both of those. थोड़ा क्यों जाएगा? पूरा अपने करेगा। साथ में लेके जा।
मैंने पहले बोला सर हेल्प करेगा
थोड़ा जाने। थोड़ा देख तो इसको घर से।
थोड़ा करना
अरे भाई जाएगा ना जरूरत ना। Make it simple. ठीक है ना 25 का टेबल लगा and 17 sorry. So now it becomes easier, no? Rather than taking decimal, avoid that.
nice रे nice phone the 4 and 1 is equal to 4. That
Because see, the number is 4, its reciprocal will be 1 by 4. If the number is 1 by 4, its reciprocal will be 4. So it becomes 2. तो चलाइए क्या बोल रहे हैं उसको
थोड़ा बैठ जा
क्यों
तैयार
नहीं ऐसे ही What he means to say is that he's got ants in his pants. That is why he's standing. खड़ा रखना. Stand straight. Stand straight. Yes. बिल्कुल सीधा खड़ा रहने का से. हाँ. देख मुखर्जी कैसे खड़ा है. ऐसे खड़ा है. मुखर्जी बाहर खड़ा करेगा तेरे को मैं बोल देता.
सर वो भी बोल रहा था.
क्या बोल रहा है वो?
सर बोल रहा जिहादी है तो बोल रहा था.
सर मैंने बोला ये बोला
लास्ट वन है.
सर ये तो गलत बात है सर. एक वर्ड को तो मेरा कर सकता हूं. 1000 वर्ड. थैंक यू सर.
What happens with 3? Fourth हो गए थे ना? अरे फोन पे लग गई. ये वाला
Sir, how 120?
ठीक है.
That's for it.
अभी Still. Over everybody.
Yes.
Question number 6. Yes, school in this one. Question number 6. Question number 6.
So 1/2 is plus 1/3. So what?
See, it says that the 3 positive numbers are in the ratio 1/2, 1/3, 1/4. 1/2 is to 1/3 is to 1/4. Whenever we are given a ratio, we generally take the common multiple. Yes or no? So we'll say let the common multiple of the 3 positive numbers be x. Therefore, one number will be 1/2x or x upon 2. Second will be x upon 3. Third will be x upon 4. Yes or no?
Yes.
फिर उसके बाद में क्या दिया है? If the sum of their squares ठीक है ना? ठीक है ना? लेकिन जब हम ऐसा करते हैं ना तो थोड़ा और ऐसा लगता है कॉम्प्लिकेट हो गया, बड़ा नंबर हो गया। मैं क्या करता हूँ मालूम है क्या? ये 1/2 है, ध्यान देना, आइडिया देखना इसका। ये 1/3 और ये 1/4 है, ठीक है? 2, 3 and 4 का एलसीएम क्या है? 12। तो इसको 12 बनाने के लिए I will consider this. Yes or no? इसको 12 बनाने के लिए और इसको 12 बनाने के लिए, ठीक है ना? तो अभी सबका डिनोमिनेटर 12 हो गया। Therefore, the numbers can be written as 6x, 4x and 3x respectively. This is clear. तो इससे क्या हो जाता है? By the given condition, 6x the whole squared plus 4x the whole squared plus 3x the whole squared is 244. Doesn't it this become simpler than taking the LCM of bigger numbers? So 36x squared plus 16x squared plus 9x squared is 244. 36 plus 16, 52. 52 plus 9, 61. X squared is equal to 244. X squared is equal to 244 by 61. 4 times. X squared is equal to 4. What is x? 2. I can take it as -2 also, but if you see, I've written there positive numbers. Samjha ke nahi? So I took x as 2. Therefore, your numbers will be 6 into 2, 12, 4 into 2, 8, 3 into 2, 6. Is this clear? Completed. Huh? ठीक है, नाइस टाइम है। तो अगले बार से सुबह 7:30 से खड़ा रहेगा, ठीक है? ओके। तो अभी
सर इसमें होमवर्क मत दो।
मैं कहाँ देने वाला था? हाँ, मैं कहाँ देने वाला था होमवर्क? थैंक यू सो मच। मैं एक्चुअली ये कम बोल रहा, मैं 200 का सोच रहा था। ठीक है, थैंक यू वेरी मच, बाय एवरीबॉडी।
I go back, please. Very mom. अबे साले मेरा पुस्तक के लिए सवाल है। आ जाओ। ये बायो की मैं भी डॉक्टर ये माया तो बायो की बहुत पसंद है अरे गंगा जी अबे
कम किया है फैन बंद कर
Good evening, good night मत बोलना. Good morning, sit down, sit down. Smart children, smart बच्चे को एक बात बार-बार नहीं बताना पड़ता है. मैं डिस्टर्ब नहीं करूँगा, मैं बैठना करूँगा. मैं लोनर बन जाऊँगा. मैं आगे बैठूँगा ना. Obviously. गले में खराश है? मैम ये करा, मैंने कुछ करा. तूने किया? प्रॉब्लम है तुझे कोई?
करने का बाय
तुम वहाँ पे बैठोगे
तो मैं जो 3 घंटे में पढ़ाने वाली हूँ 5% सीखोगे, तुम यहाँ पे बैठोगे तो 95% सीखोगे। 100% की गारंटी में नहीं दे सकती। तुम ये भी इधर बैठेगी, वो ऐसे कैसे हो सकता है?
नहीं सर, वो
नहीं मैम प्लीज ना बहुत शोर करता है मैम, हम लोग लिटरली पीछे से उठ के यहाँ आए ताकि आप एक क्रैक जोक।
तुम ये तन्मय के सामने आ जाओ, ये दूसरा सेकंड तन्मय के यहाँ पे आ जाओ, और तुम तो फर्स्ट, ये सीट तुम्हारा मेरे लेक्चर में, ये सीट तुम्हारा फिक्स है। फर्स्ट टेस्ट के पेपर्स मिले ना?
यस हाँ मैम मिल गए, मिले अच्छे हैं मैम। फुल ये मैम क्यों गया मैम? वो एफएसएस और एलएस में मेरा मार्क्स एक बार बढ़ेगा सब मिल जाएगा।
फुल मार्क्स चाहिए, फुल मार्क्स।
यस मैम आ जाएगा
29 कौन है? इधर इधर इधर
आप उसका पिटवा दीजिए।
ये क्या कर रहा तन्मय इसको पहले जरा, और ये बार बार ऐसे डिसिप्लिन मेंटेन करने के लिए हम यहाँ पे नहीं आते। स्कूल में तुम्हें ये सब सिखाया जाता है ना। नाउ अच्छा, what's your name? Last. I asked him. तुम इधर आके बैठ सकते हो? नहीं होगा? Any problem there? ठीक है, तुम यहाँ पे बैठना चाहते हो शायद, पर I don't want you to sit here. That's better, no? There. कोई तुम्हें डिस्टर्ब करेगा तो बताना। उनका डिस्टर्ब करता है? Now there. एक बात कोई नहीं करेगा ना। 5 minutes already I have wasted in maintaining discipline in this classroom, which I am not supposed to do. अब आइंदा आगे से तन्मय तुमको ये बार बार मुझे वहां से पिक करके यहाँ पे नहीं लाना चाहिए। You know, ज्यादा से ज्यादा तुम ये कोशिश करो कि हमेशा तुम यहाँ पे बैठो। Okay. We had already completed with what?
Meiosis.
We had already completed with stages of cell division done. We are already done with structure of chromosomes. Yes. We are already done with structure of chromosomes, stages of cell division. From that chapter, today we are going to finish a basic difference about meiosis and mitosis. What are some of the differences between meiosis and mitosis?
More is there.
With diagrams, I haven't done that part of the chapter, which you need to understand. Some 4 definitions, some 4 terms, 4 to 5 terms are there, which you need to understand. Before I move into that part of the chapter, if that is done, I intend to start a new chapter today. Let's see. Now, before that, I will give you a short 10-mark test right now. किसने ये exclamation mark किसने मुझे express किया? Who? How dare you do that? मैंने नहीं किया, आदत से मजबूर हो ना। I already told you this in the beginning, in my first or second lecture, surprise test. I will give you any time. जिस दिन मैं ज़्यादा खुश हूँ, obviously that day I will give you a surprise test. I don't need reasons to be happy. I know how to make myself happy. Now, always. Haploid and diploid. Haploid कुछ नहीं होता. Diploid. Now. कोई बात नहीं करेगा. First thing. 10th standard classroom है. All of you should be very serious. Next is close your books, notebooks, निकालो. In a fresh page, start writing. I will give you 10 questions, basic concept-based questions only. I am giving you. जहाँ पे तुम्हें description नहीं लिखना है. Hardly in 10 to 15 minutes you should be finishing your work. Very fair to yourself, very honest. अगर नहीं आता है नहीं लिखोगे, फिर भी इसका उसका देख के नहीं लिखोगे. Is that clear, all of you? My objective is to make you all learn. तुम भर के मुझे आंसर्स दिखा रहे हो एंड टेस्ट में यू आर गेटिंग 4 and 5. उसका कोई मतलब है? कोई मतलब नहीं है. Each one of you should get the maximum marks that you can. नहीं आता है नहीं लिखो, बट घर में जाके वो पढ़ लेना या तो अभी डाउट्स पूछ लेना. दैट इज बेटर. बट कोई भी कॉपी नहीं करेगा इधर उधर. इज दैट ओके? जो दिखाएगा उसको भी मैं अभी बाहर पूरा रास्ते पे खड़ा करने वाली हूँ. जो दिखाएगा वो भी और जो कॉपी करेगा वो भी, दोनों आराम से हाथ पकड़ के रास्ते में खड़े रह जाओगे 15 मिनट. We'll start now. You may write the questions, you may not write. Up to you. One thing before you start. Answers, if it is only one word, just write one word. Sentence frame करना, unnecessary description लिखना, please avoid. For ICSE, even if you write the correct term, one word, still you will get full marks. जरूरत नहीं है that you write a full sentence to describe or to express that answer. ठीक है? You will start from a fresh page because I'm going to correct each one of your papers. Each one of you have to show me your work. ठीक है? Fresh page से start करोगे. Books closed. Where you have written down your answers, notes of the class closed. Very fair question. मुझे ये बार-बार नहीं बोलना चाहिए. क्या क्वेश्चन लिखना है up to you. You want to write, you write. Otherwise, just pick करो and correct questions. Number दो और आंसर लिखो.
Correct is false statement.
No, only true false you write. Only true. मेहनत करना ही नहीं है, कोई तरह का मेहनत नहीं करना. Rupesh, स्ट्रक्चर तो हेड बॉय वाला है, बट सिंसियरिटी है क्या उतनी? अच्छी है तो बहुत अच्छी बात है. maximum 10 minutes. By 10:30, all of you should wind up. 10:30, 11. Subunits, not total, 4 subunits. Okay. Eight minutes.
What is the last word of
Subunits.
And is there any specific number of chromosomes that you have to show?
No, I did not ask you that. Any number you can show with 2, you can show with 3. Anything if you want. But take a lesson now. No need of doing 2. Take a lesson now. Yeah. 5 more minutes.
Diagram table.
That's right when you go, if you want to label the meaning, you want to get it. You don't label I told you I'm not trying to, I'm not going to say that I'm going to how they're selected anytime. No, put your hands. No discussion. I don't want anybody to discuss anything. Don't do contact for you. Okay. You present yourself. Hey. क्या मैं क्या सोच रहा था Those of you have already finished, take out your volume. Those of you have already finished, take out your volume. Page 26. Not to make a noise at all. Page 26. Page 26. Keep sticking some questions that I will remove. Page 26. Fill in the blanks, stick karo. No talking. Page 26, fill in the blanks, stick karo. I will again. I'm not very happy so far. मैंने जितने भी papers, जितने भी books correct किए हैं, only one student got a very satisfactory mark. Other than that, depressing. I'll keep on asking oral. I'll keep on taking oral test almost every lecture in this case. Please keep reading. Again, when you answer questions, इसका नाम क्या है? ये बैकग्राउंड म्यूजिक क्यों दे रहे थे? मैं जब बात करती हूँ ना बैकग्राउंड म्यूजिक की जरूरत ही नहीं है। ये सब तुम अपने स्कूल में ट्राई करते हो। मेरे टीचर का सो वेरी पोलाइट। किसने किया? बिना रास्ते पे खड़े कभी किए। ये याद रखना। किसने क्या बता करना है? किसने किया? कोई नहीं। एक और अगर if I catch you now, I'll really drag you to the middle of the road. Now, take page 26. Fill in the blanks. Tick करो. True, false. Tick करो. Name the following. Name the following. सब वॉल्यूम में करोगे. नोटबुक में लिखने की जरूरत नहीं है. These are all page 26. जिन लोगों का हो गया है टेस्ट, you start doing that, and others.
Will you repeat now?
Page 26. I said. Fill in the blanks. True, false. Name the following. Very short answers. And 27 में से खाली fill in the blanks. पढ़ते जाओ, कोई बात नहीं करेगा. हाँ, no. This is not a test. This is to recollect how much you remember. But I'm not that. Some of you are saying that.
उसका भी देख रहा हूँ।
No talking, no. Right?
आप चलिए परेशान कर रहे हैं
He is looking from.
मैं ये करता हूँ मेरा हो गया
Let her do some questions. I correct you. What's your name? Andy. तुम्हारा behavior मुझे ना कुछ ज़्यादा अच्छा नहीं लग रहा. I should not pick up any student and constantly मुझे उनको इसीलिए बोलना पड़ता है कि you are distracting everybody and you are extremely disturbed yourself. Please explain नहीं करो. जब हम अपने आप को ज़्यादा explain करते हैं क्योंकि गलती हमारी में रहती है इसलिए मुझे explain करना पड़ेगा ना ज़्यादा. When I'm wrong, when I'm not wrong, I don't have to explain anybody anything. अपना काम करो. I gave you enough work to do. Did I check your copy? No. Keep your copy aside. Those of you have finished writing, close your copy, keep your copy aside, and take out your volume and get started with the next work that I gave you. इतना chaos जरूरत भी नहीं है क्लास में. हाँ.
It's good.
Those of you who finished your work, keep your books closed. We check for your correct answers first. I'm sorry. हर एक स्टेज का that's why we have learned कौन सा स्टेज का क्या फीचर very very important. क्या है? तुम्हारा नाम क्या है? कितना हाँ वॉइस कितना यू आर रिप्रेजेंट मी नाउ. हाँ करेगा सब लोग बातें वही करेगा कोई अपना दिमाग यूज ही नहीं करेगा. हाँ एक जो करेगा सब बातें वही करने लगोगे.
मैम ये 4 सपाट नहीं रीडिंग 2 टाइम्स ना.
हाँ टोटल मैंने निकाले थे.
मैम पर मैम 4 सपाट
Read the question before you answer. तुम्हारे पेपर में बोर्ड पेपर में क्वेश्चन वेरी ट्रिकी. You have to read twice before you straight away answer. Wait, let me finish. Others will immediately make a change. What is your name? Anya. Who's talking? No, all in the volume you will do. Notebook में कुछ नहीं लिखना. हम ICSE की आता कौन से एंगल से मैं देखूं चलो जिस एंगल से भी मैं देखती हूँ तो भी ये ऐसा होना चाहिए और ये कैसा होना चाहिए समझ आता है ना देखो शांति से जैसे
पूरा क्या हुआ हम DNA replicates in the protein of the human cell possesses about 46 genes के बारे में बोलो 23 होगा cell division is essential for growth cell division तो ग्रोथ ही हुआ ना बच्चे
One cell division is essential for growth.
हाँ तो
गैमेट फॉर्मेशन ना हाँ गैमेट
तो
फॉर्मेशन
वो भी growth ही हुआ.
Substitution is so this is क्या problem है?
वही ना.
गैमेट फॉर्मेशन
No talking, finish your work. By the time I complete with correction, all of you should be done with your work. Did you show me your work?
liver, testes and ovaries. Somatic cells. Plate formation. Which is a process cellular where which is the process of cell division and nuclear division. And why is the pen? No, but advice because it is currently we have been wasting a lot of time. I think I gave 10 minutes to finish the work. You got more than 15 centimeters. But still see your presentation the way you write a simple word you're not able to write properly. These are the ways to write. There is no seriousness in you that is very short. This is what you continue in the next and the next lectures. I think I have to meet your parents and intimate them that what is happening with that. Otherwise, it is very shocked after some time. जब तुम्हारा performance नहीं अच्छा रहेगा, then they'll be shocked.
Now, this is the first surprise test that I took. What? Never mind. Now, this is the first surprise test that I took. Often I will take this. Please keep reading. Doubts and confusion, पूछो class में. Clarify your doubts every time. This type of test often I will take. Some of your performances are really appreciable, but some of your performances are really depressing. Now, those of you are getting, I think the highest is 8.5. How many of you got? What's your name? Aishwarya, Kirtana, Anya, Risha. Risha got 1 कम मिला कि half कम मिला. So, those of you got the highest, you should aim for the full marks next time. This is what I expect. And those of you got a lower mark, that should be a motivation for you. मुझे भी अच्छे marks मिलने हैं. Next time when I take a test, you are already in the 10th standard. ऐसा बिल्कुल नहीं है. You are not in the 2nd and the 3rd and the 5th standard. That the teacher has to tell you 1 week before what portion and what test and what date and what day. No. You should read, keep reading, concepts clear होने चाहिए gradually. Anytime I take a test, 50% marks, so any time you should be getting. On a test on any subject, 50% marks तो कभी भी मिलना चाहिए. That much should be your preparation. To get the highest and the full mark, maybe you need to revise more, polish more. That's different. So this is what I want from all of you. Now the questions that you all are solving, complete karo फटाफट. Last 2 more minutes I give you. Complete karo any doubt that you come across. Now let us complete with the answers. जिन लोगों ने wrong answer लिखा है, please correct करो. One error that I have found in most of your diagrams. बहुत सारे diagrams में most of you are drawing the cell this way. बहुत कम, I think only 1 or 2 of you have shown the furrow. Which stage it is? First of all, when you read the question, you will have to find out which stage I am supposed to draw. Now that you know that this is a stage that you have to draw, and now already I have mentioned in the question this word, so you are very much sure that this has to be there, the furrow has to be there. So this you know. Next one, what is the other error? Shandip खड़े हो जाओ भी. And what is your name? Who is Omkar? Shandip, will you please come out? Shandip, will you please come out? Get out from here! Don't try my patience, I'm telling you. I'm here to teach every student. But once you are not interested, I'm also not interested. Remember that. Get out! Come here. Get out. You have to go out of the class for 10 minutes. Otherwise, I'm not going to start anything. Come out. Come out. Give me. I don't want your sorry. Every day I've been alerting you this. All the students know the camera is on. Every day I've been alerting you. Every just today itself I told you twice or thrice. You need this now. Come on. Before I call the coordinator, please come out from there. Come out from there. I need to talk to your parents. Come out. Don't waste my time. Don't waste their time. I have to finish so many things. Come out from there. Oh, please give him way, please. Give him way. Come out. Go and inform this to our command that she's not going to take me for another 15 minutes. Dirt gone from the class. Now, next is when we talk about this, what is the next thing? Quite a number of you have made this mistake. What did you do? Okay, centrosome hai, this you have drawn all of it. Spindle bhi kiya, but many of you did the chromosome this way. नहीं, in this stage chromosome कभी भी duplicated form में नहीं रहेगा. तो कैसे रहेगा chromosome? ये जो spindle हो गया, अगर मैं एक chromosome इधर draw करती हूँ, the other one I have to draw this side.
Question में दिया है, showing the
Separation and moving towards opposite poles. तो duplicated form में draw करना ही नहीं है. ये दो error काफी सारे diagrams में था. कभी-कभी you will also get number of chromosomes mentioned. So अभी तो मैंने number of chromosomes नहीं mention किया. So you can, if the number of chromosomes are not mentioned, तो दो chromosome लेके draw करो. That's always easier. ठीक है? So the diagram does not become too clumsy. एक 4-5 chromosome मत ले लो, unnecessary the diagram begins to become clumsy, no? So draw only with 2 chromosomes. Stage name this. Breakup mark ऐसे मिलेगा 2 and 1 ऐसे रहेगा. So if the diagram is complete and correct, then only you will get to there. Remember this. Repeating unit क्या है इसका आंसर? Nucleotide. Packing unit क्या है? Nucleosome. अगर किसी को wrong मिला है, please correct करो. Gradual process or karyokinesis. Nuclear division, gradual process. Specialized structure जो cytokinesis plant cell में initiate करता है. Cell plate. Cell plate. ठीक है?
Can we write it the plant plate ना?
कौन सा?
Plant plate.
No, cell plate. No, cell plate. That's the more correct. Cell plate. This number 4. Number 4 का answer है cell plate that initiates cytokinesis. अच्छा, spindle formation responsible for separation of chromosome and plant cells. True है. Rungs of ladder of the DNA ladder hydrogen. Mainly it is nitrogenous bases supported by hydrogen bonds. तो हम nitrogenous bases ही लेंगे. Nitrogenous bases. Histone protein total of 4 subunits से बनता है 8. That's why it is called a octamer. Octamer. तो ये हो गया false. ये हो गया false. ठीक है? जो mistakes है please correct करो. Are you done with the questions of the volume, all of you?
How could you explain the second one, rungs of the DNA ladder?
The rungs of the DNA ladder, if I draw the double helical DNA something this way, nitrogenous bases यहाँ पे आता है ना, where we write A and D, or we write G and C. हाँ, nitrogenous hydrogen bonds तो है, but if I try to see what is the most important component तो nitrogenous bases ही है. If this is not there, then of course this will also not be there. So this is the most supporting structure. जिसकी वजह से वो step like structure बनता है, rungs बनता है ना, of the ladder. हाँ, these are distinctive features of anaphase. तो ये याद रखना है. Whenever you get the next type of question, this way diagram draw करना है. What are the prominent features of that stage? Prophase है, तो prophase की क्या features है? Duplicated chromosome draw करना है, nuclear membrane fragmented दिखाना है. So that way. duplication of the poles and the chromosome duplication, centrosome duplication वो सब दिखाना. Now, last 2 minutes I'll give you finish this. Any doubt पूछो फटाफट, all of you. फटाफट. I again gave you another set of questions to go through so that अगर कुछ confusion है तो clarify करो. Last 5 minutes I'll give you, then we will move on to the last section of the chapter.
Dash cells, dash is useful in establishing paternity.
Dash is
useful in establishing paternity.
Dash is useful and which one? Which one?
Question number, fill in the blanks, 4th one.
Dash is useful in establishing paternity. Into Y chromosomes. Paternity.
Not that yet. X and Y we haven't done.
You haven't done that.
Want to cut that? What is the
They may be talking about the methods also. See, there are different methods by which we can understand the paternity. There are different methods by which we can understand from the chromosome sequence. Say suppose from the blood stains, we can understand the criminals, etc. No, how we understand that process is called DNA fingerprinting. That you can write. That's the process that they are talking about.
DNA.
Yes.
Which question?
DNA.
You haven't studied. Maybe it's associated with genetics. That question is
Not in the syllabus.
no, not in the syllabus. You cannot say it's in genetics is there. It's not in this chapter. DNA fingerprinting आएगा वो answer. Number 4. उसमें process लिखो because gametes X Y.
In the textbook it is written about DNA fingerprinting, but at the top it is written not.
That's why this question has come. That's why that question has come. DNA fingerprinting.
अगले chapter का पहले chapter में ही दे.
नहीं इस chapter में है क्या DNA fingerprinting? Is there any note to it?
Page 10 textbook.
है ना? Hey. पहले से वैनिश होने वाली है
Nucleus.
हो गया डन वॉल्यूम एक्सरसाइज डन होकर उसके साथ शेयर करके करो फास्ट और डन
2 parts. Nucleus and meiosis takes place in 2 parts. Would it be cytoplasm and nucleus?
No, karyokinesis and cytokinesis.
But 2 parts to study.
Mitosis takes place in 2 parts. What are the
Nucleus and cytoplasm? 2 processes.
2 parts मतलब 2 phases. यहाँ पे parts का मतलब है 2 stages या 2 phases. You go and talk to that they. Who is that they? You go and talk to that they. Board paper में जब ऐसे questions आएगा, keep searching for that they. Who is that they who has made this paper? Are you done, Tanmay, with all? बोलो. Which question?
Dash has specific sequence of nucleotides on a
Dash has
are specific sequences of nucleotides on a chromosome.
specific sequence of chromosomes.
Nucleotides
Nucleotides
on a chromosome.
DNA. Specific sequence of nucleotide, the DNA strand. Specific sequence of nucleotides in a chromosome.
ये वाला सेकंड थर्ड वाला I'm done.
Finish, all of you finish with your work.
Yes, ma'am.
I see any disturbance in the class due to you. Straight away I'll be calling your parents. Now, let us learn some basic concepts about meiosis now. Some differences. What are some of the differences of mitosis that we have talked about? Differences between mitosis and meiosis that we had discussed the other day. Some differences of mitosis with meiosis. We are going to come across now certain stages to find out. I will explain you after that. I'm going to give you some questions. All of you should listen very carefully so that you are able to answer all the questions correct. Now, some major differences already I discussed when I taught you mitosis. No, what are some? I taught you that first of all, if I talk about the location, which type of cells show mitosis and which type of cells show meiosis. What I supposed to write? Mitosis takes place in somatic cells. What are somatic cells? Non-reproductive body cells. No. And meiosis takes place in germ cells. Reproductive cells, meaning the cells which are going to give rise to gametes. What is the second difference that we learned? The second difference is there. Second difference that we have learned is, if it is a cell showing mitosis, daughter cells form 2. Mitosis, meiosis, daughter cells form 4. What is the other difference? Mitosis is known as equational division. Why?
Because they don't
equate. Mitosis is known as equation. ये हमने पढ़ा है पिछले लेक्चर में. Mitosis is known as, mitosis is known as equational division. Why? Centrosome नहीं. क्या word बोलोगे? हाँ तन्मय. नहीं. Chromosome number remains the same.
Thank you, ma'am.
एग्जाम में जॉइंट वेंचर नहीं चलता है. ओमकार, why is meiosis known as reductional division? Why is meiosis known as reductional division? Because chromosome number that reduces to half.
48.
Whatever be the chromosome number in the parent cell, that will just get divided to half. Reductional division. Now we are going to come across that meiosis is also associated with a process known as crossing over. Meiosis is associated with a process known as crossing over. What is the meaning of crossing over? What does happen because of this? Because of the process of crossing over which takes place between chromosomes, this is what is responsible for exchange of chromatid or exchange of chromosomal segments. Exchange of chromosomal segments, and exchange of chromosomal segments is what is responsible for variation. Exchange of chromosomal segment is responsible for variation. What is variation? The new characteristics that we show when we compare ourselves with our parents. If all of you have ever realized, if you all have analyzed, that though we are or our genetic makeup is because of our parents, but even then, none of us, we are identical to our mother or identical to our father. Yes, if I talk about each one of us, we generally show characteristics similar to our parents. Sometimes the inclination is more towards the father or the paternal parent, and sometimes the inclination is more towards the mother. That means in the next concept, next chapter, we will understand not only the chromosomes, we also try to understand that the chromosomes have got section, or the DNA has got sections known as genes. That much we have studied so far. That the DNA has got sections or functional segments, जिसको genes बोल सकते हैं. But then the genes also sometimes they get expressed, sometimes they don't. So this concept in the next lecture, in the next chapter, we will learn when we study genetics. But as of now, I can only make it simple this way, saying that whenever we talk about the chromosome as a whole does not get expressed in our body. It's the section of the chromosome that is getting expressed. It is the section of the chromosome that is getting, Prisha, sit properly, sit straight. It is the section of the chromosome which is getting expressed. Now that's the reason quite a number of times we have features in our body which is quite similar like our mother, or we have got features in our body quite similar like our father. But we cannot say that we are identical even though. And at the same time, even though we share most of the characters with our parents, either the male parent or female parent, but even then about 2 to 3% characteristics that we show, our parents does not have. Our parents does not show, our parents does not exhibit. Now what is that? Why is that? What is the basic reason for this? The reason is this process, that is what we are going to study now. Meiosis generally is responsible for formation of variations in generation. Each one of us, we are born with variations. The degree may vary. But the basic concept to understand is variation always means the small changes that our chromosomes have gone through during the process of cell division. Because of these small changes, we show new characteristics which our parents does not have, does not exhibit. Quite a number of times, these characteristics may be in the form of function of the cells inside the body. These characteristics may be in the form of some external features also. And these characteristics may be in the form of our behavior also. Remember, when we are talking about the concept of characters, if I just simply go by the concept of the simple meaning of the word character, we basically understand character means our nature, our behavioral nature. That is what the English meaning of the word character is. But no, biologically the word character does not mean that. Character, if I talk about shape of the nose, that is also character. If I talk about height, that is also character. And whether I love a particular type of a sport or not, or if I love sport or music or reading, that is also a character. Each one of us, we have got a different tastes. That is also character. Each one of us, we are different in our natures and behaviors. That is also character. So now we try to understand what is variation, what is the root cause of variation. Now, one point for mitosis, I think he had already written as a difference between mitosis and meiosis earlier, and I already told you at that time, keep space, you are going to add points on your own later when we come to the end of the chapter there. So one point you had already written that the cells formed are identical. New cells formed after the process of mitosis are all identical because there is no concept of crossing over. There is no concept of crossing over, तो variation का सवाल ही नहीं है. New characters बनने का सवाल ही नहीं है. But now that I talk about meiosis, yes, chromosomes of two parents are coming together. It's an elaborate process of cell division. Chromosomes of two parents coming together, and then it is not only coming together, but they show a specific sequence of changes followed by a major change which is known as crossing over followed by variation. Now what is crossing over? How does it happen? Let us try to understand this. I now talk about a cell which is undergoing the process of meiosis. I now talk about a cell which is undergoing the processes. Now, which type of cells I'm talking about, which body cells I'm talking about, which type of organs I'm talking about.
Germ cells, testes.
I'm talking about cells of the testes, cells of the ovary. अगर plant है तो which type of cells I'm talking about?
Root, stem.
Meiosis. Cells of the flower. Cells of the flower. Anther or ovary. So I may understand that yes, the process of meiosis is happening in which type of cells in the animal and which type of cells in the plant. You get this type of questions in the paper. Now, when I talk about the chromosome, this is one chromosome that I'm starting with. This is another chromosome. So now in this cell, 2 chromosomes are there. But when I have to understand that chromosomes are in pairs, what will happen? The maternal and the paternal chromosome will always come and lie side by side. So I am starting with meiosis, and what is this phase that I start with? But I will have to call it as prophase 1. Meiosis, an elaborate phase as compared to mitosis. Mitosis is a simpler process of cell division. Meiosis happens in 2 broad phases. First phase को बोलेंगे meiosis 1. Second phase जो लगभग माइटोसिस के जैसे ही है उसको बोलेंगे meiosis 2. So meiosis 2 तो तुम्हें पता ही है माइटोसिस के जैसे ही है. Now let us learn what are some of the differences that happens in meiosis 1. Diagram drawing तुम्हारे syllabus में नहीं है. You don't have to draw the diagram, but only for understanding I am showing you some pictures. तो ये जो स्टेज है where I am saying that एक particular cell of the testis or ovary में जितने भी number of chromosomes होंगे, let it be 4. I started with 4 chromosomes here. So what is the chromosome number that I start with? The chromosome number that I'm trying to show you here is 4. Remember this. I start with 4 chromosome number. Now in this, what is happening? The maternal and the paternal. Now, why do I talk about maternal and paternal? Every particular cell has 46 chromosomes. हमारे body में, whether I talk about the cell of the liver or I talk about the cell of the testis or ovary, number of chromosomes तो 46 है. तो 46 chromosome कैसे आया? How did this 46 chromosome be obtained? How did we obtain this 46 chromosome? Only because 23 came from the father and 23 came from the mother. इसीलिए ना 23 maternal chromosome, 23 paternal chromosome. So if I have to talk about one pair, this is one and this is another. This is the maternal chromosome, this is the paternal chromosome which comes together forming a pair. So that's the reason we say that we have got 23 pairs. 23 pairs. Now, can I say that this is the 9th chromosome and this is the 6th chromosome? ऐसे होगा? क्यों नहीं होगा? Because the 9th chromosome of the father and the 9th chromosome of the mother will show a similar sequence of genes because we are of the same species. Of 9th chromosome or 6th chromosome में nucleotide sequence, now you understand, nucleotide sequence अलग होगा. So जब nucleotide sequence अलग होगा, will they have any affinity to come together? नहीं. So पहली बात ये है कि why do they come together like this, which is the first feature of meiosis, and the first feature that I write down is synapsis. इसको बोलते हैं synapsis. What is the meaning of this? Alignment of the chromosome together to form the pair. The first and the 5th. कभी आएगा पास नहीं. Why not? Because the gene sequence is different. Nucleotide sequence is different. इसीलिए they will never come to align together.
Will you repeat?
I said the first feature of meiosis जो हमने mitosis में नहीं देखा था. Meiosis, the cell which is proceeding for meiosis, first event क्या है? Which happens during the first stage, prophase 1 में synapsis. What is the meaning of synapsis? Alignment of the chromosome to form pairs. Synapsis means Aligning. So when we are talking about aligning of the chromosomes, the first and the first come together to form a pair. The third and the third come together to form a pair. The 15th and the 15th come together to form a pair. This is what is called synapses. Now, when we are talking about the concept of synapses, iske baad, we try to understand that what will happen after synapses. Now, what will be the next stage? Nuclear region hai. Now we are only trying to understand what are some of the changes in the nucleus. अभी ये जो pair of chromosome है, they will start duplicating. Prophase में duplication होता है. We had already studied in mitosis. वो यहाँ पे भी होता है. Duplication of chromosome happens in prophase. तो ये एक chromosome है. What about the other chromosome? They will also show the process of duplication. They will also show the process of duplication. Okay, let me draw only one pair. Otherwise, it becomes clumsy. I'm drawing only one pair. एक ही pair को हम अभी समझते हैं पहले. The rest is same for the other pairs also. So I pick up only one pair to find out what are some of the changes now in a sequence. तो ये वाला जो chromosome है, if I name it as the maternal and if I name this as the paternal, the maternal chromosome is duplicating. So also the paternal chromosome is duplicating. Duplication of the chromosome a major feature of prophase. इसके बाद क्या? Yes, duplication हो गया. After the process of duplication of the chromosomes, these two chromosomes, which are the duplicated copy of both the original chromosomes, इसको हम बोलेंगे non-sister chromatids. What is this? Non-sister chromatids. The next phase is in the prophase itself. The next phase is this original chromosome remains this way. And this will be this way, and then it is in the process of crossing over. It is in the process of crossing over where the non-sister chromatids have a tendency of exchanging chromosome segments, forming a X-shaped configuration. There are names for this also, but all these things are not there, that details are not there in your syllabus. But when we are talking about this, a particular stage of prophase, जिसमें if I have to name the stages of prophase, meiosis में prophase stage itself is divided into 5 elaborate phases. So we are not talking about that, but हाँ, एक stage ऐसा भी है जहाँ पे non-sister chromatids make a X-shaped configuration. But ultimately, how many chromosomes है यहाँ पे? इस stage में कितने chromosomes है? 4. ये वाले stage को बोलेंगे tetrad. This stage is known as tetrad stage. जहाँ पे non-sister chromatids crossover में रहता है. Ultimately, the entire unit has 4 chromosomes. Tetrad stage. Mitosis में आया था ऐसा कुछ? No. Mitosis is a simpler process. There is no concept of tetrad there. इसके बाद वाला phase क्या आएगा? इसके बाद वाले phase में the X-shaped configuration, they will start detaching now. First of all, they attach and make a X-shaped configuration. Actually, अगर मैं detail समझाऊं, there's an elaborate process by which they turn, twist, and then separate. The chromosomes, they first of all touch, turn, twist, and separate. तो in that process एक क्रोमोसोम को पार्ट दूसरे में लग जाता है, दूसरे का और एक में लग जाता है. That is what we talk about as exchange of chromosomal segments. तो ये कैसे होगा? If this is one chromosome that we understand, तो इसमें एक पार्ट दूसरे का आ गया. और अगर ये दूसरा क्रोमोसोम अभी पास में है, तो इसका अभी इसमें आ गया. So it is something like this. So these chromosomes have exchanged their segments. They have exchanged their segments. So simply we try to show this way. There is an exchange of chromosome from the maternal to the paternal and vice versa. This is what is we understand This process is crossing over. Crossing over is followed by a process of exchange of chromosomal segments. Now, this is the reason why we understand in complex organisms which undergo the process of sexual reproduction, we show variation. We show variation, and this concept in elaborate you are going to study in genetics. This is the reason, main reason why we show variation. What is the simple meaning of that word? New characteristics that an organism show, that a generation show, which we don't see in the parents. Though we are due to the chromosome makeup of the parents. Hamara chromosome makeup is because of our parents, but still some new characters we exhibit. Acha, abhi kya hoga? Try to remember. If this is the end of prophase, so abhi next stage would be metaphase. So abhi metaphase mein kya hona hai? In the metaphase stage, yes, the cell would be more or less oblong. The cell that will remain the same. That concept remains the same. The cell would be more or less oblong, but with a furrow. That is correct. And after that, if I now talk about that, ye ho gaya centrosome, and then the centriole, these parts are all the same. And then the spindle there. How will the chromosome arrange?
What is between the third figure?
Tetrad. T-E-T-R-A-D.
What does that mean?
What is this?
What does that mean?
What does that mean? A unit of chromosome just at the crossing over stage, जहाँ पे 4 chromosomes एक साथ है. वैसे तो if I talk about इसको हम क्या बोलते हैं? This is known as bivalent. These words are there, I think, in your book. Meiosis book part check करना. These words are there in your book. Find out your textbook. हाँ, textbook में देखो. Duplicated state of the chromosome, जहाँ पे duplicated stage में कितने chromosomes रहते हैं? 2 ना, उसको बोलते हैं bivalence. और इस stage में जहाँ पे 4 chromosomes एक साथ रहता है, उसको बोलेंगे tetrad. That is called tetrad. Okay? ठीक है? कौन सा पेज में है? देखो 16. नहीं, वॉल्यूम नहीं, टेक्स्ट बुक आंके. टेक्स्ट बुक में है. It is there in the textbook. अच्छा, we'll just finish with this. Then next is when we talk about now next what? Try to understand what happens at the end of the first phase. Will these chromosomes align in the metaphase plate? Yes, they will align together. If this is one chromosome we draw, this is the other chromosome that we draw. Paired chromosomes. So इसमें part is exchange. So इसमें part is exchange. Am I showing that these two bivalents are separated this way horizontally? No. Together they will come in the metaphase plate. So इसका next stage. इसका next. Let me finish this round, then I'll again come to what your doubt is. तो अभी ये हो गया कौन सा stage? I'm sorry. Metaphase 1. Metaphase 1. I'm showing you. Metaphase 1. ठीक है? I'm showing you metaphase 1. तो अगर ये metaphase है, and I try to show you the next stage, anaphase 1, then what should I show you? That ये हो गया centrosome, centriole. Now how the chromosomes should be arranged. Please take a note. This is one chromosome. This is one chromosome. ये हो गया the first chromosome, and now this will be the next chromosome. जहाँ पे exchange of chromosomal segments है. Is this understood or not?
So more like same in anaphase also. The
Same है, but what is the difference?
chromosomal.
हमने mitosis में ये सीखा था, जब chromosomes are aligned in the metaphasic plate, they are in the form of bivalent. Duplicated form में align रहता है. आया समझ में? If I have to show you, we'll just finish with this, then I'll give you break. Don't be bothered about the break so much. देखो. क्या? Extra time कौन देगा? अच्छा, देखो, see here. What is the difference with metaphase? यहाँ पे how many chromosomes in the metaphase stage? How many chromosomes है?
4.
The same if I have to draw for mitosis. How should I draw? ये हो गया centrosome. ये हो गया spindle. How will I draw? ये same stage अगर मुझे mitosis का draw करना है, I should have drawn this way. है कि नहीं? Meiosis में we don't draw it horizontal this way. Meiosis में rather they are very close. All the 4 will align in the metaphasic plate at once. And then they start separating, but they separate in duplicated form. They are separated in the duplicated form. So what is the first difference that we learned? If it is meiosis, pairing of chromosome happen, जो mitosis में नहीं होता है. Second difference क्या है? Crossing over. Mitosis में नहीं होता है. Third difference क्या है? If it is in the stage of metaphase, chromosomes which are aligned in the metaphasic plate is not in this form but in this form. This is mitosis. The moment we are talking about mitosis, I have to draw the diagram this way. But if I have to talk about meiosis, no, यहाँ पे एक और pair में draw कर सकती, but one pair will be this way. 4 chromosomes together in the metaphase plate. So ये हो गया third difference when you talk about differences between mitosis and meiosis. Still not over. After the break, we will continue. 2 minutes extra you will get. उसको minimize करके
क्या हुआ? चल मैं नहीं खरीदना तेरे से. अरे नहीं भाई. क्यूटी गर्ल पे मिलना.
उसमें भेज दे यार.
ऊपर करना. अरे ऊपर तो करे फिर. अरे यार
कौन सा चाहिए? हाँ यही वाला. ये वाला ये क्यों चाहिए तेरे को?
ये पढ़ाया भी नहीं मैंने.
और मिनिमाइज कैसे करते इसको पर? उधर भी थोड़े में क्या तो आया
ना भाई पूरा यूटिलिटी हो गया. अबे यार क्या नहीं भेजा
यार आंसर यार बहुत अच्छी लग रही है. हाँ वो ही लगता है.
अच्छा दिख रहा है.
खाली करो.
कोई करना स्पॉन्सर मैंने बोला है. श्रीराम देगा बाकी
चलेगा.
क्या मेरे को ये अच्छा अच्छा पहले तुम चली गई. मेरी गलती क्या है? क्या 2 मिनट रुक जाती तो कर ले जैसे.
क्या कर लेता?
अभी मैं उमेश को बोल देता यहाँ पे बैठने तो पीछे बैठ जाता बस. उसके साथ ये देखिए ये मैंने
अबे ये नहीं, ये
हमारा इसका
ये कुछ नहीं, ये काफी ज्यादा अच्छा है।
अरे इससे अगर पैसा मिलता है ना भाई अमीर हो जाते हैं।
इससे पैसा मिलता है,
इससे पैसा मिलता है
सरकार तो
वेरीफाई भी नहीं कर रहे बे 80K पे वेरीफाई हो जाते हैं। वेरीफाई हो गया ना लाइव होता है।
पैसे मिलते हैं। हाँ
हाँ
पैसे मिलते हैं।
मेरे भाई 50-50 हजार मिलता है।
ये मतलब वो है तो पागल जैसे उसने क्या क्या पता है वहाँ पे क्या रखा तो बाप ने क्या द्वारा चढ़ने का भी गिरा दी इसने हाथ से अपने
क्या बोला था आने को कुछ होता है।
हाँ
भाई होता है भाई इसको जो मिलता है उससे भाई उस दिन प्रीशा को बोला था कौन प्रीशा
मैंने उसको
कैसे कैसे लोग हैं
बार बार का बस काम मत चलाओ तो क्या नाइस होगा
चला था मैं बाहर कर रहा हूँ। अरे ईशान का बोल रहा है
ये चाहिए मान
अरे कुंडी वाली
क्या कुंडी है वेस्टर्न है ऐसा
लाइक व्हाइट डोगला
गुजराती खाना है ना मुझे बिल्कुल
डोंट से दैट ओके डोंट
मुझे
से दैट
तुम लोग मुझे कैसा लेकर चलो कैसा का खाना सकते
तुम लोग भुक्कड़ हो गए
रसम बड़ा
पता है तूने कभी राइस और रसम खाया
मैंने खाया मुझे बिल्कुल नहीं पसंद
तुझे लाइक तूने खाना है लाइक प्रॉपर केरला स्टाइल खाना है यहाँ पे
तो मुझे हाँ
खाना है
हाँ वही तो
केरला हो सकता है
नहीं उससे अच्छा
सेक्टर 1 वासी क्या आता है
मेरे को पता है यू नो अल्लम वेल्लुली एक रेस्टोरेंट है यहाँ पे क्या अल्लम वेल्लुली वो तमिल में है मतलब उसका
भाई पिछले
इंग्लिश
नवी
ट्रांसलेट
में
होगा
ये सब है मुझे पता नहीं था
नहीं वेल्लुली थोड़ा गंदा है नो
अल्लम भी वेरी गंदा है बोला
लाइक उसका ही इंग्लिश ट्रांसलेट होगा गार्लिक एंड तो प्रॉपर ऐसे पत्ते में खाना खिलाते हैं
वहाँ पे भी वैसे ही है पत्ते में
लिसन टू मी एंड देन वो प्रॉपर वहाँ पे ही शेफ्स है लाइक दैट सो गुड ना
मैं क्या अलग बोल रहा हूँ
हाँ
मेरी चॉइस घटिया है
अरे YouTube पे
वेरी गुड
अरे कोई YouTube देखता है इधर
यूट्यूब सभी देखते हैं
फनी चैनल बताना तू
अरे क्या पागल बंदा है पानी के लिए टाइम भी नहीं है अपना काम निकालो इसके वजह से बंद कर दो
यार क्या नहीं है
बच्चे हैं
अरे वो अच्छा है वो गाली देता बहुत गाली देता
वो बात सब्सक्राइब कर दे आपसे बात ना पहले अच्छी वीडियो बना
और आसिफ भी पहले आप बात कर रहे बहुत अच्छी वीडियो बना
उसका एक
अभी वो इंस्टा पे डाला
उसका ट्रेंड खत्म हो गया गाड़ी का
मुझे सिस्टर भी लगता
बहुत पैसा उड़ाता है मिस्टर
तो उसको पैसा मिलता है
उसका डबल
वही मतलब फॉलोअर का कितने सब्सक्राइबर्स हैं
और तूने ये देखा है मॉम्स इन पैरेलल यूनिवर्स
मैंने देखा
क्या
इंडियन पेरेंट्स इन पैरेलल यूनिवर्स
बेसिकली हमारे जैसे
ये देखिए वाला देख
भाई मेरे डैडी का लेंगे
अरे ये वाला मेरे जनरल
खाना बहुत मुश्किल से खाया मैंने बस सीजन तो मैंने रोज कभी मैं
भाई खाती नहीं है ऐसे हो गए ये खाना खा जाता है डिफरेंट टेस्ट क्या करता है मेरा वेट कितना है
प्रोडक्ट की बात
मर जा
मेरा कितना है 110
नहीं
69
62 62 है
62 पागल ओवरवेट है तू
ओवरवेट कैसे मैं फिट हूं मैं तेरे जैसा नहीं
भाई,
काड़ी
मेरे मॉम का वेट
ये काड़ी पहलवान है भाई
मेरा बहुत वीडियो है
भाई ये 70 प्लस है
मेरे मॉम का वेट 68 है
इससे पता चलता है इसकी मॉम को कितना खाना देती है भाई
भाई ना भूखा नहीं मरता
अरे
है
हमारे
भूखा मरता है भाई भूखा मरता है
भूखा मरने का ही नहीं है
ऊपर से जो
मेरे घर पे क्या है ना मेरे घर पे क्या खाने का क्या नहीं खाने का मेरी मम्मी डिसाइड करती है
जो सामने आएगा वो मैं खा लूं मैं तेरे दिमाग में
ये ऐसा कुछ नहीं हमारे घर पे तो मॉम तो बनेगी वही बाहर का मंगाना होगा ना तो मम्मी बाहर ही वरना मम्मी को ऐसा मतलब प्लीज करना पड़ता है ज्यादा खाता है
नहीं अभी तक भी नहीं पड़े लेकिन हफ्ते में 4 दिन फिक्स
बाकी के 3 दिन क्या खाती है
मेरे को भी बनाती है 4 दिन बाद
हमारे घर पे
अगर बाहर जाना होगा जरूरी खाना है नहीं
भाई ये तो एक दिन ना जाते जाते रास्ते पर बेहोश के जाएगी तो अकेले आना
जब मम्मी मेरे को बोलेगी ना लड़कियां अच्छी पढ़ाई करके ये फोटो
तो फिर तेरी मम्मी बोलेगी क्या सारे क्लास में लड़कियों की फोटो निकालते रहते हैं
तेरा भी फोटो है मेरे पास भाई तेरा भी कहां वाला तेरा भी है ये वाला
क्या है क्या करेगा तू
अरे का फोटो है मेरे पास अरे का फोटो
धन्यवाद नहीं वाह
क्या करते भाई घर पे यहाँ पे घर पे चुपचाप
लेके आ रही है छोटे कुछ साथ साथ और ये अंदर लेके बाहर ले
उन्नति यू नॉट उन्नति तू वहाँ पे बैठ रही है
तूने तो करना है
ये कश्मीर बन रखा है
ये कॉरिडोर के एकदम
तू है तेरे को भी
एकदम
आने वो लड़कियां कहीं ना अभी और गिर गए उसमें से
बहुत ठंडी है ये तुम लोग ठंडी नहीं
अभी ह्यूमिडिटी हो रही भाई पानी आ रहा है हाथ पे इतना ठंडा अबे भाई पानी हो
ठीक है तो तुझे बैठ
हां ठंडी तो पानी और भी
गर्मी लग रही है
अरे रुको भाई अब मैं तेरी फोटो लेके आ रहा हूं अरे यार रुक जा मेरी अबे भाई अबे मैं सोच रहा था क्या बोल रहा है अरे यार भाई कर लो
सितंबर में वो टेस्ट दे रहे हैं Mom is very cold. Please tell me
all of you. Why is this coming? क्या है ये? Now, so what did we study? We have created not a noise.
एसी का मतलब बड़ा नहीं हो गया।
ये लोग सब इस तरफ क्यों आ गए तुम लोग? हाँ? Why did you go at the back? क्यों? एसी ठंडा लग रहा है।
दो मिनट
Rita, you will get up from there and come here. Get up fast. Okay, so what we were discussing was, when we talk about these different phases, what we are now trying to understand that if this is meiosis 1, for the concept of meiosis 1, after the process of first stage of meiosis now, after the first stage of meiosis, let us just try to follow complete with the process of cell division and then find out what should be the number of chromosomes. So now in this case, when we are talking about the two new cells formed, how should I draw the number of chromosomes? How should I draw the chromosomes? There will be in one cell, the chromosome will be something like this. Yes or no? In one cell, the chromosome would be something like this. In one cell, the chromosome would be something like this. And in the other cell, so the chromosome is moving in the duplicated form. So in the first phase of meiosis, that is anaphase 1, did the centromere divide? No, the centromeres did not divide. But did we not study about centromeric division as a major event when we talk about anaphase of mitosis? Yes, always. Always. So whenever we are talking about anaphase of mitosis, during anaphase of mitosis, there is always centromeric division. But in the first phase of anaphase, I'm saying anaphase 1 agar hai, is there any centromeric division so far meiosis? No, there is no centromeric division. And now if I follow this with this cell and all the stages again, meaning this cell will again go through all the stages, what will be the later stages. This will go through a stage known as telophase. Of course, telophase 1 we have to write. Then what? Then it will again start with prophase 2. So that is how it happens. Telophase 1 over, meaning one full cycle of meiosis is done. Now the next stage will be prophase 2, metaphase 2, anaphase 2. So if you are following, if you are able to understand, then when I talk about all these stages, prophase 2 ho gaya, then what should come? Then metaphase 2 aayega. This will be followed by what? Anaphase 2, no? this will be followed by anaphase 2. And now if I have to draw telophase 2, what will be the number of chromosomes? I'm just following this cell. I'm just following this cell, and in this cell, what will happen to this duplicated form of chromosome? It will separate, no? Yeah. It will separate. And when it separates, now this chromosome comes in one, and in the other cell, one chromosome. And if I follow this cell, am I not going to get two more cells? I'm getting two more cells, because from here I'm getting two cells. If it is mitosis, same like mitosis, I said. Meiosis 2 is same like mitosis. And I'm talking about meiosis 1 jab khatam hua and meiosis 2 jab shuru hua. That's why we write prophase 2, metaphase 2, anaphase 2. And यही cell को हम follow करते हैं. We don't know what are the stages. कौन से stage में क्या होता है. So at the last stage, I'm talking about now telophase 2 now. I'm talking about telophase 2. So telophase 2 में how should I draw the stage? Duplicated chromosome फिर से split होगा. In which stage? Anaphase 2 में split होगा ना? Centromere will split and one chromosome एक cell में आ गया, दूसरा chromosome opposite cell में आ गया. And now if I follow this, the same thing is happening. I'm talking about this chromosome and यहाँ पे दूसरा chromosome आ गया. But हाँ, in one chromosome there will be this exchange of chromosome. ये भी रहेगा. At the end of meiosis now, how many cells we got? 4 cells or 4 gametes, because I'm talking about reproductive cells. 4 cells or 4 gametes, and the chromosome number reduced to half. Chromosome number reduced to half. Because we started with 2 chromosomes, and after that what happened? The alignment of the chromosome happened later. So we started with a cell which had only 2 chromosomes, and then we started with a cell which had only 2 chromosomes. Remember, I followed with only this. I am following only with one pair. So 2 chromosomes, no? So the parent cell is having 2. So how many chromosomes the daughter cells are having? 1 each, no? Is this clear or not? How many of you not clear? How many of you not clear with this concept? क्या doubt है दुर्गेश? सुनोगे तो समझोगे. I said I will again say this. After this, I'm going to give you 5 questions. So all of you should write this correct. I am starting. पहले मैंने 2 pair of chromosomes दिखाया था, but I started following in the diagram एक ही pair को मैं करती गई. So now I'm not confusing you. I'm only showing you 2 chromosomes. जो एक pair बनता है, जिसमें एक maternal chromosome था और एक paternal था. This is coming together. Coming together को क्या बोलते थे? Aligning और synapses. उसके बाद each chromosome will duplicate. ये तो प्रोफेज में कॉमन है. Each chromosome duplicates. अगर chromosome duplicated chromosome जहाँ पे 2 chromosome एक साथ रहता है, उसको बोलते हैं bivalent. पर meiosis में कुछ additional होता है. That is after the process of duplication, the non-sister chromatids always cross. The non-sister chromatids always form a cross-shaped configuration process, जिसको हम crossing over बोलते हैं. Cross joining भी बोलता है इसको. So that may be termed as cross joining or crossing over. अब crossing over के बाद क्या होना है? Exchange of segments. पैटर्नल क्रोमोसोम का एक पार्ट मैटर्नल में लग जाएगा. मैटर्नल का एक पार्ट जाके पैटर्नल में लग जाता है. जीन सीक्वेंस सेम रहेगा अभी? नो, जीन सीक्वेंस अभी थोड़ा चेंज हो गया. That is the reason we start showing new characteristics. क्योंकि अब तक तो ये पैटर्नल और मैटर्नल क्रोमोसोम था. अभी ये सारे क्रोमोसोम्स जो हमारे जाइगोट में हमारे सेल्स में आना है, is it same like the chromosomes of the parents? No. ये सब का जीन सीक्वेंस और इसका जीन सीक्वेंस अलग होगा ना. So these are the cells that we are going to have when the gametes are undergoing the process of fusion, fertilization. So ultimately these are the chromosomes we are going to get in our body, no? So ultimately now next what? Crossing over जो हो रहा था वो separate हो गया. exchange of segments. अभी ये this entire tetrad 4 क्रोमोसोम एक साथ मेटाफेज प्लेट में अरेंज होगा. अब यहाँ पे how many chromosome? This is 2 chromosome. Duplicated condition है, but how many chromosomes? What is your answer? What should you write? How many chromosomes you are trying to show here? 2 chromosomes. जो duplicated condition में 4 दिख रहा है. Actually है तो 2 chromosome ना. Then अगर ये metaphase है, तो anaphase में क्या होगा? In duplicated condition they split. But centromere कहीं से भी split हुआ है? No. Centromeric division does not happen in anaphase 1. तो only the 2 bivalents जो duplicated condition में है, एक duplicated cell एक तरफ और दूसरा duplicated cell दूसरे तरफ. So that is what has happened. तो ये अगर anaphase है, तो जब telophase होगा and cytokinesis होगा, one cell will get one duplicated set. One cell will get one duplicated set. Now, if I say R chromosomes or R chromosome number is 46. R chromosome number is 46. So, इस cell में how many sets should be there like this? 23 sets. Understood ना? तो यहाँ पे 23 sets होगा. यहाँ पे भी वही से. अब ये जो cell है, it will follow the same process like mitosis. मतलब if I start drawing the stages of this cell, it is the same as mitosis. मतलब ये duplicated set को फिर हम metaphase plate में लगाएंगे, फिर anaphase होगा. Now centromere divide होगा. अब centromere divide होगा. Centromere divide हो रहा है इसीलिए now that I follow with all the stages. अब जब इससे which is a stage of I'm showing telophase. तो telophase के बाद prophase 2, metaphase 2, anaphase 2. जब telophase 2 आएगा, this pair of chromosome has become split up to form 2 single chromosomes. तो एक single इस तरफ गया, एक pole में जाएगा. The other single will go to the other pole, ultimately forming 2 cells by cytokinesis. तो एक सेल से 2 सेल मिला और ये दूसरे सेल से और 2 मिलेगा. Ultimately, we get 4 cells. This was our starting cell, है ना? This was our starting cell, and we had been dealing with all these stages. Gradually, we have come across what are some of the stages, main stages to find out the differences. तो ये तो हमारा parent cell था. अब यहाँ से यहाँ हम compare करते हैं. Parent cell how many chromosomes? 2. Daughter cell में how many chromosomes? 1. Is this reduction division? हाँ. Chromosome number is just reduced to half. Is it following the rule of meiosis? हाँ, because parent cell है और 1 से 4 cell बन रहा है. 4 cells, 4 gametes. 4 gametes. Now, a little bit more that we have to study with the concept of meiosis is the last concept that we try understanding is that when we now look into the process of meiosis always results in the formation of the gametes. So, abhi I may try understanding that ये जो cell है, this particular cell, if it is a cell of testes, if I take it as the cell of testes, then how many gametes will it produce? It will give rise to 4 cells. Each cell will have 23 chromosomes. So this I may talk about as male gametes. Or if I am talking about this as a cell of the ovary, how many gametes should it produce? 1 functional gamete and 3 smaller, almost non-functional gametes, but the number of chromosomes remains the same. Now, what do we understand by this? I say that ये जो है, if I have to talk about in the concept of meiosis, when the cell of testes undergo the process of meiosis to form gametes, हाँ, 4 cells तो बन रहा है. All the 4 cells have equal amount of cytoplasm and chromosomes. इसमें भी 23, इसमें 23, इसमें 23, इसमें 23 और चारों में the amount of cytoplasm is also the same. Is it the same when I talk about the cell of ovary? No, not that. In the process of meiosis, talking about the cell of ovary, one cell would be having the maximum amount of cytoplasm और बाकी जो 3 cell बनेगा that will have a very small amount of cytoplasm, scanty cytoplasm रहता है. So I may summarize this way that if it is a cell of ovary which undergoes the process of meiosis, cytoplasmic division is uneven. Even नहीं होता है. But what about the nuclear division? No, nuclear division even होता है. Nuclear division is even. Nuclear division is even. So that means that when I talk about nuclear division, हाँ चारों में 23 chromosomes रहता है, but cytoplasm will not be the same. इसके बहुत सारे significance हैं, we'll learn of course later, but just now to mention, this is the cell which after the process of fertilization is going to form the zygote, and only by the process of fusion of these 2 chromosomes.
ये किसने किया है था मार्किंग
क्या है
आपने ले वो
Now, when these 2 gametes undergo the process of fusion, what is also known as fertilization, we get to see a zygote. So how is the chromosome number coming back to 46 again? By the process of fertilization. One gamete with 23, the other gamete with 23. Now the chromosome number will be 46 again. What is the process I'm talking about here? Fertilization. What is the outcome of fertilization? Zygote. A single cell known as zygote. So in this chapter till here, in the next chapter when you study reproduction, you will learn a lot of detail about it.
Why is it that only one like cell has more?
That is how our body is made up. Because the significance is why is this process? See, the body is made up in such a way when we talk about the meiosis process in cells of the ovary. The cytoplasmic division always is uneven, where we get to see one big cell is getting formed and the rest 3 small cells getting formed. The reason is the body is built up in such a way perhaps because this is the cell which is going to form the zygote later, because in the process of fertilization this 23 chromosomes only will enter this. The cytoplasm of this cell is not going to be used up. अगर ये सेल भी इसके जैसे छोटा होता या तो if all these 4 cells would have had an adequate amount of cytoplasm, perhaps after the process of fertilization nutrients उतना zygote में होता ही नहीं to carry out the process of cell division. And before this zygote, once in the process of cell division, the zygote gets converted into an embryo. Just to give you the basic concept, ये embryo में उतना amount of nutrients रहना चाहिए, cytoplasm रहना चाहिए, so that it can support the process of cell division for a long time before it gets attached to the uterine wall. So, if the cell would have been all of the same size, तो शायद ये process possible नहीं होगा. Any doubts? We are done with this chapter on cell division, structure of chromosomes, cell cycle. We have already studied the 3 main sections of this chapter. We had already studied with 3 main sections of the chapter. This was the last topic that you have to understand. Take 5 questions and we'll wind up with this chapter now. Notebook, all of you take out your notebook, write 5 questions from the last segment of the chapter and we'll be done with this. Any doubt, any confusion that you have, please clarify. Any doubt, any confusion you have, clarify. Now the first question, right? First question,
this is also closed.
No, not a problem. You can see from the book also one. Right. The first question. Define. First question. Define a diploid cell. Define a diploid cell. Full stop. Next part of the same question. Define a diploid cell. Next part. What is the diploid chromosome number? What is the diploid chromosome number of human cells? What is the diploid chromosome number of human cells? Same question. That's the second part. Number two. Okay. This X-shaped configuration, the name is there in your syllabus. The name is there. This X-shaped configuration को बोलते हैं chiasma. Chiasma. The X-shaped configuration form is known as chiasma. Right on the next question. Right on the next question. What is the significance of? What is the significance of? What is the significance of crossing over? What is the significance of crossing over during meiosis? Shandip, can't you speak properly? Why do you always stoop like this when you sit? Sit properly and write on your own. Why are you looking into his notebook? You are not supposed to miss. You are supposed to have that much of concentration in the classroom. Your behavior is really getting into my nerves now. What is this? What I told you. What is the significance of crossing over during meiosis? Number 3. You may leave 2 lines, 2 or 3 lines. If this answer should not be more than 1 to 2 sentences. इससे ज़्यादा लिखना ही नहीं है. Number 3. Mention the chromosome number in human gametes. Mention the chromosome number in human gametes. 2 types of gametes. No, human produces 2 types of gametes. Chromosome number in human gametes. Number 4. What are homologous chromosomes? This I will give you the answer. What are homologous chromosomes? What are homologous chromosomes? What are homologous chromosomes? This you should know.
It is logos and logos.
L-O-G-O-U-S. L-O-G-O-U-S. What are homologous chromosomes? Next, last, number 5. Mention one difference. Mention one difference between anaphase of mitosis and anaphase 1 of meiosis and anaphase 1 of meiosis. That is it. 5 minutes में complete करो. अगर कुछ doubts है, confusion है, पूछो, clarify. So this is the last segment of the chapter. We are done with this question. Complete करो. All of it. No talking. Nobody will talk. Very seriously, you will finish your work. करो fast. You will not copy down without understanding. What is the question I gave you? What is the significance of crossing over? मतलब crossing over से what ultimately leads to crossing over? Crossing over से क्या benefit होता है? Crossing over results in the formation of variation. Crossing over, crossing over results in exchange of chromosomes, exchange of chromosomal segments and variation. Variation दिखना ही है. Crossing over results in exchange of chromosomal segments and variation. Homologous chromosome, when you come to that, I will give you the answer. That's a proper definition that we should write. Now we had already studied this. Listen here. When you have to write about homologous chromosome, this is what is homologous chromosome. What did we learn? I showed you this is one chromosome and this is the other. What I told you, this is the maternal chromosome, this is the paternal chromosome. That is the reason they are coming together and they are of the same number. The ninth or the ninth will come together. The fifth or the fifth will come together. So what are you supposed to write? You may try writing on in your own words also. Understand what it is? Homologous pair of chromosome. What does that mean? In our cells, actually all pairs are homologous. So what is the meaning? A pair of chromosome which consists of a maternal and a paternal chromosome. और इसको actually homolog बोलते हैं. Homolog. Simple, you can write a pair of chromosome which consists of a maternal and a paternal chromosome. एक क्रोमोसोम मैटरनल रहता है, दूसरा क्रोमोसोम पैटर्नल रहता है. That's the reason. और it has got a special name, homologous pair. Why the word homologous? Why it is not heterologous? क्योंकि 9th and 7th कभी पेयर नहीं बनेगा. 9th and 9th pair बनेगा, 7th and 7th pair बनेगा. That's why homo. Homo means similar. A pair of chromosome which has a maternal and a paternal chromosome.
And which has
a maternal and a paternal chromosome. A maternal and a paternal chromosome. Done. What is the doubt, Unga? Diploid chromosome number in human cells 46. 46. That's a diploid. What is the definition of diploid? इसको याद है तन्मय? What is the definition of a diploid cell? Q.
Is the cell which has two half sets of
chromosome. Two half sets of chromosome. अगर haploid होता one half set. What happened? What is the doubt? क्या doubt है? ये मैंने intentionally ये दो questions मैं फिर से लिखा रही हूँ because why talking students should write down the number of chromosomes in human gametes. 23 pairs. 23 लिखना है. Pairs कहाँ? Gametes never have chromosomes in paired condition, no? श्री राम तुम्हारा मैजिक शो हो गया तो are you done with the question? Tanmay, are you done with the answers? चलो फटाफट इंट्रोड्यूस नहीं करना है.
I'm getting the answer. Yes, ma'am.
What is the question?
I mentioned one difference between
Write down. Write down. Write down. Write. Thank you, ma'am. तुम बड़ा speedy बोलते हो. But if you don't get full marks, I break every bone that you have in your body. Again, we say. क्या बोला हमने ओमकार? आंसर लिखो. During anaphase of mitosis, division of centromere takes place, or splitting of centromere. That's a more appropriate word. Splitting. Division या splitting, any word of course you can write. Centromere.
That is the point, right? For
during anaphase of mitosis, division of centromere, splitting of centromere takes place. Full stop. क्या हुआ?
spelling of centromere.
किसको नहीं आता है centromere का spelling?
किसको भी नहीं
Other side, anaphase 1 of meiosis, or during anaphase 1 of meiosis, during anaphase 1 of meiosis, there is no centromeric division. There is no centromeric division, and bivalence. You now know what is the meaning of bivalence. Bivalence move to opposite poles. Bivalence move to opposite poles. ठीक है? Sit straight. Done? All of you are done with this?
Yes, ma'am.
So genetics I don't intend to start now. Let us take a break from this concept of chromosomes and genes. क्या हो गया?
we have to discuss the unity volume.
अच्छा. have only one more. Besides, go ahead. Now the volume answers, I had given you this exercise to solve, no? All of it. Take out your volume. Take out your volume, page number 26 and 27. I asked you to solve. 26 and 27. All of you, take out your volume. Come to that page. Page 26. I'll start from Shriram then. Start. The first, no volume. Start now. Sit down, sit down, sit down. Fill in the blank. The first three answers follow.
Chromatin fiber is made up of DNA and
Histone. Next.
the chromatids are attached to each other at centromere.
Centromere. This is spelling. Histone. 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 Number 3.
Base stands are made up of phosphate, pentose, sugar, and 4 types of nitrogenous bases.
Bases. Very good. Then I continue. Number 4 say DNA replicates in the
interface of the cell.
DNA replicates in the dash of cell cycle. DNA replicates in cell cycle के कौन सा phase में? S phase of cell cycle. S phase of cell cycle. हम मना आज मुझे trophy.
मैंने भी प्रोफेसर
Cell division नहीं है ना? You did not ask me cell stage लिखना है कि cell cycle में? You did not ask me. DNA duplicates in which stage of cell division? Meiosis के which stage में? Prophase. But वो formation of the DNA actually if I look into interphase जो prophase के पहले आता है, वो already in the S phase में initiate हो जाता है. Completes in the prophase.
नाम पर पीछे चेक कर देना इंटरफेस देखा.
कहाँ पे?
So the answer is in stage of
No, interphase में कभी नहीं होगा. DNA replicates in the dash of cell cycle. When you are studying the entire concept of cell cycle, the entire thing is one interface. But यहाँ पे ये पूछा गया है कि इंटर सेल साइकिल की कौन स्टेज में, कौन सा स्टेज में, तो इंटरफेस कैसे लिखोगे? इंटरफेस तो लिख ही नहीं सकते क्योंकि जो हम सेल साइकिल पढ़ते हैं, we actually started the interface stage only in detail. The interface stage can be divided into parts like the G1 and the S and the G2 and then M. तो अभी वो G1, S, G2, M में कौन से phase में duplication starts? Happens. S phase, sympathetic phase. S phase में. चलो तब मैं continue with answer the next also.
Human cells possesses about that.
Human cell possesses about 30,000. Normal 30,000. More than 30,000, so approximately 30,000 you can write. Chromosomes 20 Listen here, Survesh.
Sorry, ma'am.
I mean, becoming more naughty here.
ये belated birthday वाला बहुत क्या नाम है? Survesh.
Sorry, ma'am.
ये sorry और thank you को बड़ा sweetly बोलते हो, और तुम्हारा मार्क्स effective होना चाहिए. I am such a person who never speaks sweet and I don't appreciate the sweetness in somebody. मुझे खाली results चाहिए. That's it. Results नहीं मिलेगा, goals तुम्हारे वैसे भी मैं crack करने वाली हूँ. Next is. So maybe answer you gave me now. Human cells, okay. Number 6, cell division is essential for growth. Cell division is essential for growth, maintenance, repair. वैसे तो बहुत सारे, but majorly अगर एक common word लिखना है तो growth लिखना है. Continue now. 7. Fatapat. I don't want anybody to talk when we are discussing the answers. So, mistake किया है तुमने? Erase करो, लिखो. Number 7. Come on.
Media or bacteria divide to produce similar independent cells by mitosis.
Very good.
Meiosis takes place in the reproductive organs like testes and ovaries in humans.
Correct.
Mitosis occurs in somatic cells.
Testes and ovary in humans. दो word है ना, दो gap है ना. Next.
Mitosis occurs in somatic cells while meiosis occurs in germ cells.
Germ cells. Okay, all of you got the answers now correct. Come to the next section. True, false. You don't have. True, false.
In cell divisions occur for three studies growth replacement repair. The number of chromosomes is the same and is true.
In cell division, this sentence is little bit incomplete. In mitotic cell division, make that sentence correct. In mitotic cell division, ये सारे features you can summarize for mitotic cell division. बायो क्लास के बाद उसके साथ बैठ के उसके हेयर का टेक्सचर को भी थोड़ा रिलैक्स मिलेगा. Next. क्या हो गया? उनका को मैसेज करना है.
Just shut up.
I don't want any noise in this class now. Continue.
4 phases of mitosis are similar in both animal and plant cells. True.
4 phases, major 4 phases. Correct. Next.
In animal cells, cytogenesis occurs by plate formation. False.
False. True, false. All of you got it?
Yes, ma'am.
Yes,
Come to the next. Name the following.
Within the process of cell division, when nuclear division occurs, cytokinesis.
Correct. Next.
Within the process when cytoplasmic division occurs, cytokinesis.
Next.
Between two divisions, when cells are in resting conditions, the phase is called interphase.
Interface. All of you got
What
it?
is the second one?
Cytokinesis.
Correct.
Cytokinesis. Come to the next. Very short answer time. Bolo, please.
Name the type of bond that links nucleotides of two complementary DNA stand horizontally edge bonds.
Nuclear horizontally. Correct. Next.
Name the three components of a nucleotide: nitrogenous bases, pentose sugar, and phosphate bond.
Okay.
How many histone molecules are there in a nucleosome? 8 of them.
8 molecules. Okay.
Name the 4 nitrogenous bases of DNA: adenine, guanine, thymine, cytosine.
Next. Lisha. Name the biologist who
Name the biologists who had explained the double helical structure of DNA.
Watson and Crick. Next.
Why new cells need to be produced? Growth. The new cells need to be produced for growth, repair, and gamete formation.
Kirtana, not to disturb her.
What are the main phases of mitosis? Interphase, prophase, metaphase, anaphase, telophase, and cytophase. What is telophase?
Done. Done.
No, ma'am, 2 questions are not.
Acha, next is what are the main phases of mitosis? You are writing all in a sequence. Interphase से लेके telophase. Done, all of you. Now next is what is telophase? No, you are writing interphase, prophase, metaphase, anaphase, telophase. That's it. Okay, cytokinesis, karyokinesis यहाँ पे नहीं लिखना है at stages. Next. What is telophase? What is telophase? For 1 mark, what to write? A stage of cell division. Now the prominent event you have to write there. You don't have to describe so much. What is telophase? A stage of cell division in which prominent event क्या है telophase का? Nuclear membrane reappears and chromosomes uncoil. Furrow depends. ये है. Any two event if you write that is more than enough. Nuclear membrane reappears. Nuclear membrane. Spindle fact that is not a very significant thing. सबसे significant क्या है? Uncoiling of chromosomes. तो फिर से thread जैसे बन जाता है. Uncoiling of chromosomes, reappearing of nuclear membrane. Nuclear membrane reappears. Uncoiling of chromosome. That's it. अच्छा ये हो गया ना. Now फटाफट. Come there. Fill in the blanks. Shape of the DNA molecule was studied by Rosalind Franklin. Histone DNA from a complex called nucleosome. DNA. I'm sorry. Dash are specific sequences of nucleotide of a chromosome. Dash are specific sequences of nucleotides of a chromosome. हाँ, genes भी लिख सकते हो. Correct. Genes भी correct answer है. DNA segments of DNA, no? Genes are. Genes are correct answer. हाँ? The question is saying dash are specific sequences of nucleotides on a chromosome. So if I say the entire chromosome which is made up of DNA के sequences को हम genes बोल सकते हैं. So that also that's a more perfect answer. वो लिखो. Genes लिखो. Genes are specific sequences of the DNA or the chromosome. Okay. Come to the next one. Dash is useful in establishing paternity. I said DNA fingerprinting और पे लिखना है. Number 5 बोलो. Number 5. Sex cells are formed by meiosis. Mitosis takes place in two parts. Karyokinesis and cytokinesis. ये लिखना है. कौन सा? Sex cells are formed by meiosis. During pairing of chromosome in meiosis, the dash chromosomes come to align side by side. Homologous. And the last one, what is it? The last is the dash are surrounded by radiating rays. What is surrounded by radiating rays? Centrioles are surrounded by radiating rays. Are we done with the exercise? All of them? Done. Any doubt anybody has? Any question? Any confusion? So we are done with this chapter. As I said, I'm not going to continue with genetics right now because in none of your school this chapter is done, no? Genetics, this is not done in any of your schools, so we don't start with this right now. And we'll take a break also from this concept. If you are going out, go fast, come back fast.
It is not in our course.
Can I open and see?
What?
Ma'am said cell will be in all the
But वो portion के PDF में नहीं है.
Cell.
पढ़ना पड़ेगा.
The first chapter. It's not reading.
Can I just open? Now the chapters that we can start would be this. I think these chapters are done, no, with your absorption System
Absorption done? In none of your schools, absorption, transpiration, these are done? No?
This is the
Tell me.
Ma'am, we can start excretory systems.
we have absorption by excretory system, endocrine system, transpiration, structure of chromosome, and population. Population
Self study was higher.
Tentative
Ma'am, you are starting with excretory, right? Yes.
Excretory actually circulatory के बात करते तो अच्छा है, but let's see.
अब school में भी होता है like that.
अरे don't talk about your school. Your school they start. No noise in the class. No noise in the class at all. So we start with the next chapter, that is excretory systems. So if that is done in your school, we can start with that. Excretory system as the next chapter that we are starting with. I don't want anybody to talk. Every 5 minutes, I should not mention that in the classroom, no? That is what should be done. That is what I do. That is what we do here. But since you need this chapter more as per your school requirements, so not a problem. When we talk about the concept of excretory system, let us start with this. What are we studying here under the concept of excretory system? What do we understand by the concept of excretion? How do we define this word? Excretion. Excretion, how do we define this word? Yes. Removal of nitrogenous waste. Now, whenever we are talking about the process of excretion, we more or less try to understand the process of filtration of the blood. And by the process of filtration of the blood, the waste in the liquid form, which contain a large amount of nitrogenous substances, has to be released out. So that is what we understand as the process of excretion. You are asked, define excretion. Write down the answer. Define excretion. What is the proper definition that you write? Define excretion. Start from a new page. Write down the name of the chapter. The first definition that we start with. Excretory system. The release of waste substances. The release of waste substances in the soluble form rich in nitrogenous substances or rich in nitrogenous compounds. Rich in nitrogenous compounds. Rich in nitrogenous compounds by the way of kidneys. By the way of kidneys is known as excretion. Is known as excretion. Now, when we are talking about excretion, let us also try to first of all understand that what are some of the primary substances that needs to be released or removed out from our body. What are some of the substances, primary substances which may be toxic for our body if they are retained? That is only the nitrogenous substances we are talking about. There are quite a large number of other substances. We will talk about the nitrogenous base. There are some other different type of substances also which needs to be eliminated. So first, that is not excretion. When you talk about undigested food, that is not a part of excretion at all.
Water.
Excess of water, yes, excess of water. So, surplus of salts and ions. Otherwise, we understand that yes, the ions and salts are so much necessary for our neurons, for our nerves. But surplus of it, as it is gets released out. That last group will not talk unless and until you are participating in the conversation. You will not start talking. Otherwise, ये चारों को चारों सब बाहर जाएंगे. You will only talk with reference of the chapter and the discussion that is happening in the classroom. उसके अलावा, now this is the last warning to this 3-4 of you there. Straight away, I'm going to put all of you outside the classroom. ये खाली स्टार्टर था 5 मिनट 10 मिनट का जो डिस्प्ले मैंने अभी दिखाया. I'm very famous for putting children straight away out of the classroom. घंटे भर खड़े रहोगे, अगले दिन पेरेंट्स के साथ आओगे. I have been doing this always. Not a new thing for me. Discipline you have to maintain in my class. ये तुम्हारे schools में ये बहुत lenient behavior teachers follow करते होंगे. Here we will not accept all this. Now, so what I said, what are some of the substances that needs elimination? Which are some of the substances that needs elimination? And what would happen if the elimination does not take place? First of all, to talk about carbon dioxide by the way of lungs. So what would happen if the carbon dioxide does not get a way to eliminate? If the carbon dioxide is remaining in the blood for a long duration, what would happen if the carbon dioxide does not find a way to get out of the body? The muscles are not getting adequate amount of oxygen. In that case, energy supply would be less and the blood becomes venous. The blood becomes impure. The blood starts becoming venous. So the carbon dioxide which is released from the tissues, what is that process called? You had learned in the last years, cellular respiration. Very good. So when we are talking about the concept of respiration, जो पिछले साल सीखा था. So with that concept, when the cells and the tissues of our body which are constantly releasing carbon dioxide, by the way of blood, it should reach the respiratory surface. Though we are talking about elimination here, are we talking about elimination of carbon dioxide by the way of the kidneys? No. Why not? Because carbon dioxide cannot get released or carbon dioxide is not present in our blood in such a form which can be dealt so far the process of diffusion at the level of the kidneys, but can be dealt so far the process of diffusion at the respiratory surface is concerned. वैसे तो kidney tubules में भी diffusion होता है, same like the lungs, but the process and the method is absolutely different than what we study in the alveolus, in the lungs. So first I say that yes, carbon dioxide is a toxic compound formed from the tissues. This also shows a process of diffusion to get eliminated, but why not by the way of the lung? Why not by the way of the kidneys? Why by the way of the lungs? Because it has something to do with hemoglobin. It has something to do with hemoglobin. So by the way of hemoglobin, it can be dragged, it can be brought to the respiratory surface. It cannot go to the excretory surface, something known as the lungs, or something known as the kidneys. What about the next? When I now talk about nitrogenous substances, how are they formed? How are they carried? When I talk about nitrogenous substances, something to very clearly remember, where are they formed? And what is the different forms that can be possible in organisms? What is the most toxic form that is getting formed in our body? So if I'm talking about nitrogenous forms, remember the excess amino acids. Now amino acids, you know, no? What are amino acids? These are the components of proteins. Otherwise, if I say, I would say that proteins are made up of amino acids. So हमारे body में, when we understand the process of digestion, yes, the blood is loaded with nutrients, no, after absorption. The blood which is loaded with nutrients, and by the process of circulation, it also reaches the liver. In the liver, this excess amino acids undergo a process, and this excess amino acids undergo a process known as deamination. And by the process of deamination, where certain new compounds are formed, but during this process also we come across that urea is a compound which is formed as a byproduct. Where is this cell? Where is this process happening? This particular process is happening in the liver. This particular process is happening in the liver. Nitrogenous waste, where are they formed? In the liver. Nitrogenous waste, from what type of substances are they formed? During the breakdown of amino acids. During the breakdown of amino acids to form some other compounds. I may also talk about, they may also result in the formation of carbon dioxide and other different type of compounds. Complex compounds are there. So whenever we are talking about that, basically you are to understand right now that where are they formed, how are they carried. These type of nitrogenous compounds, majorly when we talk about urea, is formed in the liver due to the breakdown of excess amino acids. But yes, when we talk about this urea which is present or formed in the liver cells by the way of circulation, now when this urea is carried to the body tissues, it is extremely toxic if it is retained in the body tissues for a longer period of time. So this urea needs immediate elimination. But this urea again, by the way of circulation, has to reach the kidney surfaces to get diffused at the level of the kidney tubules and the blood vessels. So this urea, we are a terrestrial organism. This is the most toxic form of nitrogenous waste formed in our body. But other than that, there are other different type of nitrogenous waste formed in our body in small amounts, which I can talk about as uric acid. In our body, uric acid is also formed. In our body, uric acid is also formed, which is a toxic nitrogenous waste. But this uric acid is formed, may get deposited even in our tissues. That's the reason they need to be eliminated. But this is not formed in our body in large amounts. If I have to talk about the reptiles, etc., etc., these are those group of animals where the uric acid is formed in a large amount. For us, urea is released in a large amount comparatively. The other type of nitrogenous waste to just name would be ammonia. But we don't have any tissue in our body where ammonia is as it is released. But if I have to talk about aquatic organisms, yes, ammonia is released in large amount. But in our body, yes, ammonium compounds in small amounts may be there. But that is not a major type of a nitrogenous waste. So actually there are three types of nitrogenous waste, and we belong to a category of organism where in our tissues, this is the primary nitrogenous waste formed. Sometimes you will get questions in your paper. Name the two primary nitrogenous waste formed in our body. You have to write urea and uric acid. The two primary nitrogenous waste released in our body would be urea and uric acid. Since I am talking about what are some of the substances to be released and by what way, what is the other type of nitrogen? What are the other type of substances that we can talk about? Now, if I talk about by the way of the liver again, another type of substances that needs to be released out of the body would be bile pigments. Another type of substances that needs expulsion, that needs elimination would be the bile pigments. अभी ये कौन से pigments है, कहाँ से आता है? Where are they formed? They are again found in the liver. And how, from where? The breakdown of hemoglobin. This hemoglobin always breaks up into some component pigments, simpler form of pigments. And what are these pigments called? These are called bilirubin and biliverdin. Where are these reactions taking place? These are also taking place in the liver. This is also taking place in the liver. And this biliverdin and bilirubin will further get broken down to give rise to another type of a pigment, and this is what is known as urochrome. So when we talk about the biliverdin, bilirubin, first of all, these are obtained by the breakdown of hemoglobin. These reactions are very much happening in the liver. And to talk about the digestive role also, they generally impart a color to the undigested food, etc., etc. That's a different part. But urochrome, now that I talk about, yes, urochrome is a pigment which we often are aware. This is what is responsible for the light yellowish color of the liquid waste. Urochrome, to remember this word. So indirectly, we can also say that urochrome is formed by the breakdown, or it is a breakdown product of hemoglobin, because first of all, hemoglobin gives rise to bilirubin, biliverdin, which further gets broken down to give rise to urochrome. What is the significance? What does it lead to? Gives color to the liquid waste. Gives color to the liquid waste. And the fourth to talk about would be excess salts and water. Excess salts and water. What are the primary salts necessary for our body? Can you tell me? Sodium chloride, or rather the sodium and the potassium salts, very much necessary, Kirtana and सुबह ग्लूकोज पी के आया करो, या तो ग्लूकोज बॉटल में भर के लेके आया करो, हर 10 मिनट में पीते जाओ, you'll get lots of energy, the body will be heated up. सुंदर दिखो ना, अरे ये सुबह उठ के करने का काम है, अभी नहीं. मिस्टर तन्मय समझ गया ना? इंटेलिजेंट पीपल को खाली इशारे की जरूरत होती है. नाउ व्हेन वी आर टॉकिंग, अरे किसने बोला? पहले तुम अपने बारे में सोचो. एनी टाइम तुमको मिलने वाला है. Now, what I said, the fourth. Biliverdin. Deamination. That's a process. Then DEAMINATION. Amino group remove hota hai, that is why it is called deamination. Now we come to this excess salts and water. How do we get excess salts and water in our body? By the way of food, by the way of water. Water also carry a large number of ions. So when we are talking about the variety of ions and salts, of course they are very much necessary. Why are they necessary? For the formation of different type of pigments in our body, for the formation of different type of enzymes, hormones in our body, these ions and salts are necessary. But of course, we have these ions in surplus. In surplus. So these ions have to be released out of the body. These ions have to go out of the body. These ions, which are there, how will they get eliminated? In the dissolved form again, by the way of water. So again in the kidney, when we talk about the process of filtration of the blood by the way of the kidney, there also we are going to understand how the excess amount of water can be released out, and they carry a lot of salt. They say the salts and ions are very necessary. Again I say, but yes, excess salt. Can you tell me what is the other way by which small amount of ions and salts and water make their way out of the sweat? Perspiration, sweat. So by the process of perspiration, the excess amount or a small fragment or a small fraction, I should say, a small fraction of the salt, ions, water can be removed out. But to specify, we also have to remember the function of the sweat glands and the skin in this process. No? So we are talking about by the way of the skin glands, by the way of the sweat glands of the skin, this process can be fulfilled. So now, if I talk about that, what are some of the substances to be removed out? Yes, this becomes primary. Of all the different type of waste substances of our body form from time to time that needs elimination. What is the primary? Primary is this. इतना क्या ठंडा लग रहा है?
I'm sorry, you were just saying.
Ma'am, fan on the
Ma'am, this is not yet in this class. Yes, ma'am.
मैं जो पीने भी तो कबाब बन जाएगा तो मैम सबसे डिस्ट्रैक्शन हो रहा है
Go and get the remote. Okay. So now, what are the main substances that needs released from the body? Always to remember, your answer always should be nitrogenous bases. Name the primary organic compounds to be released out. Name 2 primary organic compounds to be released out. Primary nitrogenous substances to be released out. Your answer is this. Always to remember, your answer is this. Urea, uric acid. Ammonia, I already told you, ammonia would be a major nitrogenous base if I'm talking about an aquatic organism, not for a terrestrial organism like us. We cannot write ammonia as a major nitrogenous base formed in our body because ammonia release requires a large amount of water, subsequent water release. But we are terrestrial organisms, we cannot release so much of water. Otherwise, we will always suffer from dehydration. We don't. You will learn in the next section of the chapter that how our kidney is designed, and it always tries to conserve water in our body as much as possible, giving away the waste in a soluble form with minimum amount of water loss. अगर हमारे body में ammonia main product होता, then every time we would have suffered from dehydration and शायद हमें gallons and gallons of water पीना पड़ता हर बार. So that not necessary. पहले ये सीनियर सिटीजन लोगों को पहले तो कुछ इलाज दो इन लोगों को. ये सीनियर सिटीजन को इतना ठंडी लगती है. हाँ. I'm going to sweat. I'm going to run out of the class.
I'm going to go to washroom.
You are going to go during the break. I allow 1, I allow 2, and then the entire class is break. Nobody will ask for this during the class. Then next is we start discussing about the excretory structures, the primary excretory structure, that is the kidneys. The primary excretory structure, that is the kidneys. Take out your notebook, all of you. Take out your notebook, all of you. Pencils. You will have to draw diagrams about the excretory system. Here are 3 diagrams you are supposed to know. Excretory system and the parts function. Section of the kidney, parts function, and the structure of the nephron. We start with this, the first one. Excretory system of human. We are supposed to draw. I don't want any noise. No noise in the class. No noise in the class at all. All of you start drawing the diagram. Only with pencil. Nobody will use a colored pen. Nobody will use any sketch pens to draw and to label. Only with pencil you will do, all of it. Drawing, labeling only with pencil. When you draw the diagram, remember your diagram should come in half of the page. Your diagram should come in half of the page, not full page. Your diagram only should be drawn in half of the page. Put your diagram little bit towards the left side of the margin so that all the labels you can put on the right side. For this planning, you have to remember every time when you draw the diagram. Number one, only with pencil. Number two, diagram almost in half of the page to complete. Three, start your diagram little bit towards the margin. If this is my book, I will not draw the diagram on the right side this way. I will put my diagram here so that I can put all the labels on the right side. There should be space left. Remember this when you do. Right. This is the one kidney is not limited, but one should be little bit at the lower level.
It should be a little above.
The right should be little bit lower, the left should be little bit higher up. So that slight difference, not a major difference also when you show the diagram. A slight difference only. No talking. Kirchana and this girl, Prisha, stop talking. तुमको भी साथ में भेजेंगे.
Draw that or the blood vessels and stuff.
Artery vein you have to show, no? Every time the vena cava you have to show in the
Ma'am, in the school teacher told that if you go inside, you don't have to show all of that. If they're asking you to draw the kidney, ureter, and urinary bladder.
When you show the excretory system, the blood supply, you need to show artery and the vein. You will be asked also this type of diagrams in the paper. You will have to use the same type of diagram you will get in your paper also. Diagram based questions you will be given the same diagram. Pencil should be dark. Pencil should be dark and sharp when you draw the diagram.
And that
You will draw all with pencil. I am drawing with different colors so that I can mark to make you understand. You will not use any color. I don't have a blue color, so I am using this green color. जो परेशान करेगा सीधा बाहर सिंपल हाँ You should know the relation of these blood vessels when you are drawing the diagram. This type of questions you will get. Label the parts 1, 2, 3, 4. You should understand which vessel is connected with what. The blood supply when we talk about.
Splinter.
Splinter. Splinter is a Finish up all of you. Don't scale, just freehand you are supposed to draw. Free hand you are supposed to draw.
Ma'am, Prisha is asking if this is the art class.
From the today, I'm in school, ma'am, we didn't draw the face.
School
In school, that red and green makes it. Ma'am, we have drawn.
Renal vein, renal artery, you have to draw.
Ma'am, we have to draw.
When you are asked to draw the diagram of excretory system, if you don't draw, that is okay, but you have to know the renal artery, renal vein, because diagram based question will come with this two vessels always there. When you get a 5-mark question on the diagram, at that time you always have to label this part. Aorta के साथ renal artery and vena cava के साथ renal vein. The relation you have to remember. So I will not teach you without the vessels. What your teacher is doing, that is up to her. What I teach is my way of teaching. How much you want to take, that's again up to you. The diagram of the excretory system without the blood vessels, no, we never teach you that way. You have to know the relation of the blood vessels with the kidneys. Done, all of you? Finish all of you. We did with pen.
It looks so artistic.
So this will be there for your diagram drawing. You will get this, draw the diagram of the excretory system in human. For 3 marks, you get this diagram drawing where 2 marks would be for drawing, 1 mark would be for labeling. The next class we are going to learn the functions of each of these parts and then the section of the kidneys. We are yet to learn another 2 more diagrams here, the section of the kidneys. So first of all, today we finished with the excretory system. So just have a look here and you can finish the diagram at home also. What are the parts we are looking into? The primary excretory organ. What is it? Kidneys. From the kidneys, the tube-like structure. What are these? Ureter, which drains the liquid waste into the muscular organ known as the bladder. The ureter and the bladder, they are made up of smooth muscles, remember. But when we are talking about the opening up, the opening of the bladder, which is known as the urethra, on the inner wall, they have got some specialized muscles. Remember the name? It is known as urethral sphincter. And this sphincter will control the process of release of the urine. That's a particular word, we'll learn that. Finish this diagram at home, all of you. Complete the diagram, all of you. Many of you haven't finished.
को याद है फुल गाना अभी होमियो यूरिया हो गया अभी वो खून मुझ पे खून रहा होता है उसका था ना वो मुंह में खून निकलता अरे वो खून निकलता उसका नाम क्या होता है अरे पढ़ा है बे बायो में अरे मुंह में खून निकलता है पढ़ाया था ना क्या था
इसमें क्या
कंडीशन में
हाँ अरे
अबे वो